fact that we have had a great last couple days, and all the people that went out last night and enjoyed the evening. You can hear how Leo and I's voice have changed by the beginning of the meeting till the end of the meeting. I hear there was some burger run toward the end of the night. There's only one time per year I dance to the Backstreet Boys, and it tends to be end of September. So thank you for allowing that to happen to everybody. So that was truly awesome. So we have a great finishing program lined up, and we're focusing predominantly on antibodies, and then there's going to be a little competition toward the end. So we get to hear first about how donor-reactive antibody causes graft injury. We'll go in from the science to the clinical applications to the histology, and then we'll get into the clinical aspects. So Rob Fairchild, I will bring you to the stage again. Thank you very much. And we'll hear about antibodies.
Okay, thank you, Alex. So what I'm going to do is I'm going to give a very didactic lecture about some of the things we know about how antibody mediates graft injury, and then I think we'll have plenty of time after that to go through a little basic science on that. Okay, so I don't have any disclosures. So one of the things that we're realizing now with the advent of the new or the immunosuppression strategies that we now use is we've decreased the incidence of T-cell mediated rejection, and now we're seeing more antibody mediated rejection at all years after transplant, and actually antibodies appear to be the major cause of graft injury and graft loss now. And we need to understand why that is. So the first thing you need to realize is that anti-donor MHC antibodies or anti-HLA antibodies have both direct and indirect effects on epithelial cells and endothelial cells. Endothelial cells are usually what they react with first, and the leukocytes that mediate this intense inflammation during antibody-mediated injury. And it's the consequence of that inflammation that is the primary cause of antibody-mediated graft injury outside of a culture well. A lot of the initial experiments were done in vitro. We're now moving toward more in vivo type of approaches to understand that. Okay, so during acute antibody-mediated rejection, I'll just show you very quickly what we're talking about in a clinical sense, and then Michael Mengel will elaborate on this, is that when antibody binds to the graft endothelium, we get this microvascular inflammation with margination in kidneys of monocytes and neutrophils and macrophages into the peritubular capillaries. That's a sign that there is antibody-mediated rejection ongoing, and also staining for evidence of antibody binding to the endothelium through the activation of complement. And I'll talk quite a bit about that. And we can detect the presence of complement split products that bind to proteins in the vasculature of the graft by staining for C4d or C3d. Okay? Whoops. Okay. So, and this is just the same thing happens in heart grafts. When you stain, you can see, again, C4d, the presence of C4d indicating complement activation, indicating antibody binding to the circulation in the myocardial capillaries. And you can actually see the macrophages going into the capillaries of the myocardium by staining by CD68. Up here is the H&E of this. So you, again, one of the main things is that antibody binding recruits leukocytes and activates them at the site. And that is actually what I'm going to show you is the cause of injury in this case. Okay? Again, The effects of the antibody are mediated through activation of complement. There is instances where antibody does not activate complement, or there is no evidence of C4d binding. I'm sure Dr. Mengel will tell you about this. And then there are direct effects of the antibody on the endothelial cell. I'll show you how antibody binding to the surface of its target molecules on the surface of the endothelial cells activates those endothelial cells, and there is direct effects of antibody on endothelial cell proliferation. So this would cause chronic injury and the vasculopathy that we often see during chronic antibody-mediated injury. So the first thing we're going to talk about is complement activation. Complement can be the cause of hyperacute rejection. So in this case, this was an ABO incompatible graft that was transplanted to a patient. This happens within about 20 minutes. You get the binding of the antibody to the surface of the endothelial cells in the graft, which express the ABO carbohydrates. IgM antibody that the recipient has binds to those carbohydrates, and you get very rigorous activation of complement, and the graft just fails almost immediately with thrombosis and just shutting the graft down. Okay, and this just shows what happens when you have that you have this IgM binding in the graft and this really intense activation of complement in the graft. So complement is a series of enzymatic steps from the complement proteins that interact with the Fc portion of the antibody, which is bound to the target. It exposes the site and sets this activation of these complement components in process. And then there are these series of amplification steps where different enzymes are generated. They do other things as well. And then in the end, what you get is formation of a pore so that you lyse the cells. The contents of the cells leak out. Now, I will tell you, while I'm talking about this right now, is that we evolved this system to fight against bacteria, and that's what happens when bacteria do this, or are bound with antibody like this in the presence of complement. Okay, so that's what we've evolved this system to do. We can do this in culture too. When we put antibodies against endothelial cells, epithelial cells, we can get this formation of these pores and lyse the cells. In vivo, in a graft, we don't get to this point, and one of the reasons we probably don't get to this point, or rarely get to this point, is because these cells have regulatory proteins that interfere with this process getting to this point. So again, the injury is caused by that microvascular inflammation of recruiting the leukocytes, more than making these holes in the endothelium and epithelium. So what you should know is that there are different properties of the different immunoglobulin isotypes, IgM and IgG. IgM is a strong complement activator, and IgG, it depends on the isotype of the IgG molecule, whether it does or not. Here are some other functions, comparisons of those isotypes, but what you really want to know is that antibodies that bind complement have stronger association with antibody-mediated rejection than those that don't. Okay, so here are the isotypes in a series of patients that had antibody-mediated rejection, looking at the isotype of their, or the properties of the antibody that are mediating the, in the DSA, mediating the injury. Those that activate complement are more associated with the injury than antibodies that do not activate complement. Okay, so complement has a lot of functions. That's what we've evolved the system to do. They can be chemoattractant, so they chemoattract or direct the trafficking of these leukocytes from the circulation and from the bone marrow to the site where they're activated. They can be cytokine-like. They have adhesion molecule properties. There are receptors on different cells that function within this. And I've talked about pore formation, and we're not going to talk about that again. Okay? But the thing is, again, you start out here with complement binding to the FC portion of the antibody molecule, and then it amplifies. So these enzymatic steps become stronger as you go through this cascade from C1 to C9. Okay? And that there are fluid or soluble factors that are released from the complement during this process of this cascade. These enzymes clip out certain proteins from the complement complex, and they're released into the fluid, and they have certain functional properties, and they're also portions that bind to the cell, and they also have certain properties. Okay? So, for instance, the fluid part of complement that is released during complement activation can bind to receptors on neutrophils, macrophages, all of these different leukocytes, and even T cells and B cells, and it has some activation signals that it mediates into these cells. At the same time, when complement fragments are bound to the cells, these are different fragments, there are these cells too that have these receptors for these bound components, and you can get adhesion, chemoattraction to the site of the activation. So again, you don't need to know the specifics, but please realize there are fluid forms that are released into the inflammatory milieu. There are bound forms that are bound to proteins at the site of the activation, and they both have functions. So again, the first component, C1q, binds to the antibody, and that begins the activation of the complement cascade. All you need to know is that as these enzymatic processes go, there are split products. Some are released again, fluid phase. So you have this amplification, and you can see there are more and more. It's enzymatic. It just generates these complexes. At the same time, then, the cell that is being that the complement is being activated on starts to upregulate expression of molecules which try to regulate this, okay, called complement regulatory proteins. And I'll go through that very briefly. So they try to put the kibosh on it. It's not completely effective when you have a lot of antibody that is a complement activating antibody. And finally, this amplifies to the point where if you don't have these complement regulatory proteins, you get this pore in the cell, and you get lysis of the cell. And again, that is not seen very often. Okay? So this is a statement from just what I said from this consensus conference, that you have this intravascular thrombi, interstitial thrombosis, you have some necrosis and things like that, but you rarely encounter endothelial cells that are necrotic at the site of antibody-mediated rejection that is mediated by complement, because, I'm saying it over again, they have these regulatory proteins that inhibit the complement cascade from going that far. Okay? So here are some of these regulatory proteins right here. Decay accelerating factor, a lot of work has been done on this, and some of these other factors. These are the kinds of things that if you work in pathology or you're looking at antibody-mediated rejection, you're going to be become familiar with. And so these factors do different types of things. They either dissociate components of complement from the complex as it's forming, so dissociation decreases this amplification, and there are some like factor 1 with a couple other membrane-bound factors that actually cleave the components of complement and inactivate them. Okay, so the whole thing about complement, again, is that when you start to get a pro-inflammatory response with complement activation, you are going to activate the expression and function of these regulators, such as decay accelerating factor and some of these other ones, that are going to target these very strong pro-inflammatory effects of this complement cascade and try to attenuate it. Nevertheless, as it's activated, one of the things that is going to happen is that you are going to still have this intense leukocyte infiltration in this microvascular inflammation. Okay? And this is just, again, pictures showing complement activation in a heart transplant. Okay? So I've told you this, they're fluid and they're abound and they recruit, and different leukocytes have different receptors for different components. You don't need to remember this, I won't test you on this. But when you have these membrane-bound complex fragments from this complement cascade, then these receptors can bind to these various components, and it helps them adhere to the surface of the endothelial cell, and it also activates the macrophages or the monocytes, the eosinophils, along with other things that the endothelial cell is expressing because it has this bomb going off on its surface, okay? And so this is just a picture or a depiction of the mechanism of these things that diffuse complement. So there are direct effects of antibody on endothelial cells too. Elaine Reed has been one of the pioneers in looking at this. When you bind anti-HLA antibodies onto the surface of endothelial cells, you transduce a series of signals through the mTOR pathway that causes the endothelial cells to become activated. They produce cytokines and express various things I'll talk about in a minute, and they start to proliferate. Okay, and again, that's where we get vasculopathy from those, at least it's thought, and those, we need more science to go on that. There are also direct effects of antibody on the endothelial cells. When you bind and you get these signals, the endothelial cells have these little things called Weibel-Palade bodies, and they're filled with von Willebrand factor, which is a component of the coagulation cascade, and they also have this molecule called P-selectin, which is something that functions in slowing leukocyte rolling on the surface of the endothelium. So what you get is, and this is just a figure showing how fast this happens when you activate the endothelial cells with anti-HLA IgG, and you can see here in vivo that now you get P-selectin on the surface of the endothelial cells. Okay, and again, I think some people heard yesterday Leo or Marissa talk about how cells slow down on the surface of the endothelial cells through interaction with P-selectin and then undergo diapedesis, okay? The other thing that can happen is that binding of the antibodies can stimulate the production of chemokines by the endothelial cells, so that is important for activating the integrins that are required to stop the leukocytes on the surface of the endothelial cells. So again, antibody binding to the endothelial cells is doing all these things to recruit leukocytes to the surface of the endothelial cell and to get them to stop on there and to diapedese into the interstitium and cause this inflammation. So I've told you about the effects of this on cell injury, and most of this has been done in vitro cultures, And in the last 10 minutes, I'm going to talk about this. We have not had good animal models to study this from start to finish. And most of the data that we have has either been done through in vitro studies or from clinical observations. And people like Michael Mengel and his associates and Annette, they have been instrumental in taking clinical observations and trying to put them into a model. But it's been very hard to try to get a model where you can actually look at this starting from A to Z. And this is a lesson for the fellows here that if you keep your eyes open, sometimes good things will happen. We were looking at molecules involved in leukocyte trafficking to grafts, and we stumbled over the fact that if you use mice that are knocked out in CCR5 as recipients of heart or kidney allografts, the antibody response is dysregulated. They make 50 to 200 times more antibody, and they will reject kidney grafts. And when you reject the kidney grafts, the pathology is identical, or mostly identical, to that which you see in clinical antibody-mediated rejection of kidney grafts. And this includes that margination of monocytes and macrophages and neutrophils in the peritubular capillaries, and you get this glomerulopathy with fibrin deposition into the glomeruli, and you actually get, sometimes the pathologists look for this double contouring of the glomerular capillaries, and that's there as well. Okay, so in this model, you get very rapid production from zero, DSA production, up to it reaches peaks about day 14 or so, and then it's maintained until the graft's rejected. I'll show you that right now. So one of the weird things in mice is that if you put an MHC mismatched kidney graft into most strains of mice, you get very low amounts of rejection of those grafts, and they go out to day 100, 150, they have some chronic injury, but the mice cannot acutely reject grafts. In the CCR5 model, which is these two groups here, they acutely reject that because they make antibody. And if you get rid of the antibody, and we've done it several different ways, then you get these kind of results. Okay, so you have to have antibody to mediate rejection of these grafts. The other thing is there's a time between reaching or achieving the peak antibody or a DSA titer and the time that you get failure of these grafts. This is a graft survival model. The only kidney that's left in after, say, day 7 is the allograft. So when the allograft fails, then the mouse dies and suffers. So I just want to go back to some clinical results, and these were studies that Luis Hidalgo and Phil Halloran did in Edmonton. They were looking at trying to achieve a molecular microscope of kidney graft injury, and they saw that when they had early rejection episodes of kidney grafts that looked like pathologically it was mediated by T cells, they had one signature, and when they had late rejection that looked like it was associated with antibody-mediated rejection, they had a signature that was associated with NK cells, natural killer cells, which I don't think we've talked too much about. But these are a kind of a cross between innate and adaptive immune response that is really important in fighting viruses and tumors. Well, when we looked at rejecting grafts in the CCR5 knockout model, lo and behold, at the time of rejection, we got the same genes that expressed that Luis and Phil are seeing that are associated with NK cells. So we asked what the role of NK cells are, and we used an NK cell depleting antibody. Now we get about 50% or so of the grafts surviving long term. They look pretty good, at least till about day 100. And when we look in the grafts, we can see NK cells infiltrating the graft, and they start proliferating within the graft, and they start expressing this molecule called CD107 that means they're cytotoxic. I will say one of the things you should notice is there aren't that many in there, and the thing about NK cells is they are proliferating in there, but they just go from cell to cell and mediate injury and inflammation. So this just shows the proliferation of the NK cells. So these data strongly indicated that NK cells are involved in this process, like is thought in clinical transplantation. Now the thing, we needed more proof of this. I have 5 more minutes. So one of the things about NK cells is they usually don't attack cells because in addition to these activation receptors right here, I guess it's right here, they also express these other receptors that transduce inhibitory signals that are dominant over the activating signals, okay? And what these inhibitory signals bind to is self-class 1. So when you have a viral infection or a tumor, they will downregulate class 1, and so the NK cells then can be activated through the activation receptor. So what we hypothesized is that if we expressed recipient class 1 on the graft, then we shouldn't be able to get acute antibody-mediated rejection. And to do that, we just made an F1 of the donor and the recipient. We took the kidneys, we put it in the CCR5 knockout. Again, the hypothesis is by expressing the class 1 of this, we should not get acute antibody-mediated rejection, and that's exactly what we see here, okay? Now, one could say, well, you've diluted the expression of the allo class 1, but we get the same titers of DSA in response to those F1 grafts, and we do not get NK cell activation, indicating or suggesting that mediating inactivation of the NK cells during antibody-mediated rejection through engagement of this inhibitory receptor abrogates acute antibody-mediated rejection. So, however, there's a lot of antibody in there. What is it doing? So we took these grafts that are out past day 60 and we look at them, and lo and behold, we see what we see in the clinic. We start seeing interstitial fibrosis, we see glomerulopathy, and we see arteriopathy, which is right here, okay? So the antibody, if it doesn't have NK cell activation, it can mediate acute antibody-mediated rejection, but eventually, over time, it's beating on the endothelial cells of the graft and the other cells in the graft, and you'll start getting this chronic injury. And I don't have time to talk about those pathways, but this is the end. The results of this mouse model are consistent with the clinical results. They suggest that NK cells are needed and synergize with the DSA. They're activated during this process to mediate acute antibody-mediated rejection. I'll show you how that works in a minute. When you don't have NK cell activation, and you could think that this happens by immunosuppression as well. So maybe if you're taking your immunosuppression and you make antibody, you may not have activated NK cells to do this, and you'll eventually develop that chronic injury rather than acute injury. So the way we see this working, and I don't have time, as I said, to show you all the steps, the DSA binds to the MHC molecules on the graft. It induces these pro-inflammatory cytokines. It induces chemoattractants, chemokines for NK cells and monocytes and neutrophils and other cells. It also induces the expression of this molecule called RAY1, which is the ligand, and it's the only one we know of, and we have a knockout mouse for it now, that binds to this NK cell activation receptor. There are many NK cell activation ligands. This is the one that's induced, at least in our hands, by this DSA. So what happens then is the NK cell is activated, it enters the graft interstitium, monocytes as well do, and the monocytes have to produce this enzyme called myeloperoxidase that's used to kill bacteria that it ingests, and that is involved in the activation of the NK cell. And there's also a source of IL-15 produced that causes the activation of the NK cells within the graft. We think it's these cells, but we don't know for sure yet. But when this happens, you get acute graft injury, and when you inhibit NK cells, these macrophages and probably neutrophils cause chronic injury and cause the stimulation of myofibroblast generation. Okay, so if you want to hear more about this, I'm going to actually talk, I think, for 45 minutes or something about this at this meeting, and you should all sign up. It's going to be a great meeting and a lot of science like this, and I think that's it.
There's a couple clinical observations that we want to try to correlate with your pathophysiologic descriptions here. So one is with the complement-mediated injury that you described, how do you explain or what do you think about the use of eculizumab as an effective treatment for antibody-mediated injury, as it's C5 through, it's essentially blocking later stage?
Yeah. So I guess it seems to work a little bit,
yeah.
But this is probably one of the most powerful innate immune systems there are, because we evolved not to have infection.
Yeah.
So, it may be partially effective. I think we need better antibodies to do that. Yeah. And I think it's important that they're so proud that they have one of the most expensive drugs on the market.
Fair enough. So the other correlation, or the other observation, we toss around the word clinically, accommodation, especially when we talk about ABO incompatible versus anti-HLA. Can you maybe conjecture about the difference or the C4D staining or this complement deposition that might be different in an ABO scenario than a HLA antibody scenario?
Yeah, so I think when you're starting to get intense complement activation, you're going to activate the endothelium, and I think you're going to get injury, and the endothelial cells are going to do bad things. Yeah. So when you have accommodation, I would predict that you have to stop the process pretty early in the whole activation system.
Okay. Yes?
So Laurie West has been working for a long time on this issue of ABO accommodation. And for a long time, it's true, people have argued that the ABO can induce accommodation and anti-HLA antibodies by virtue of its ability to signal into the endothelial. Cell tends to not accommodate for, permanently or for a very long period of time, and ABO can. But I think that there is new data from Laurie West that says that actually there is no true accommodation in the sense that the endothelial cell becomes resistant to damage. But what it does is that it changes its glycosylation pattern a little bit differently, so that even though by hemagglutinin the antibodies come back, but they're not identical. And so it's not as pathogenic. And I think, Ibrahim, if I'm wrong. So I think that the issue of whether you can get stable accommodation as a means to sustain lifelong graft survival, I think it's now much more uncertain.
So what's the definition of accommodation?
Basically, I think that we're, again, using the word accommodation to say why is there normal kidney function or normal graft function, yet we have evidence of antibody-mediated deposition. So no inflammation, graft function, no graft dysfunction, essentially.
Presence of circulating animals.
Correct, yes.
And I'm happy to announce that at the next year's ATC in 2020, there will be an ABO incompatible plenary session, so stay tuned to that.
Well, with that, you can keep standing and continue on. So thank you very much, Dr. Fairchild. Yeah. All right, so we're moving on to the next phase of our physiology and immunology, which is the detection and identification with HLA typing of sensitized patients. Annette Jackson from Duke. Thank you.
Thank you all for coming. You were quite impressive last night, so I think you can handle HLA. So these are my disclosures. 3 take-home messages in case you have to leave early. HLA matching went out of vogue with the introduction of calcineurin inhibitors, but I'm happy to say that HLA matching is back in vogue. I want you to understand the difference between CPRA and DSA, especially when you're reading older literature, when they seem to mix the terminology. And finally, when we're looking at DSA and its risk potential and its injury potential, MFI tells us important things, but is only part of the story. And so my first and only polling question, I want to know how many alleles you think there are of A2. So how many flavors of A2? 10 to 50, 100 to 500, or greater than 700? Not A alleles, just A2 alleles. All right, so you do believe that HLA is polymorphic, and most of you said 100 to 500, but I'm here to tell you that there are over 877 different alleles of HLA-A2, and that's 100 more alleles than when I gave this lecture last year. So that's called job security where I come from. So HLA is a very unique genetic system. It is more polymorphic than any other genetic system, and in fact, more than half of the alleles do encode different proteins, and so that's also unlike other polymorphic loci in the human system. And we began DNA typing in the 80s, and this was through an international collaboration during the 11th International Workshop, and we shared reagents, and we identified many more alleles through different populations. Sequence-based HLA typing came into vogue in the 90s, and we discovered even more alleles. And now with the rollout of next-generation sequencing, I'm afraid you're going to see that this slope of this curve is going to even accelerate further. Why is HLA so polymorphic? The purpose of the HLA molecule is to sit on the cell surface and display peptides to the adaptive immune system. We need it to be polymorphic so as the pathogens evolve, we have enough HLA molecules that will always be able to present a pathogenic peptide to the adaptive immune system. None of us in this room, unless you're related to one another, are likely to be HLA identical, and so with this type of diversity, it is unlikely that a pathogen will be able to take us all out simultaneously. As Rob mentioned, some viruses subvert this surveillance by downregulating HLA on the cell surface so they can fly under the radar. However, NK cells are constantly probing cells to look for the missing HLA. And when HLA is missing, it doesn't engage the inhibitory receptors, and then the cells are lysed. So all of the current HLA typing methods use polymerase chain reaction to amplify the loci of interest. In the middle of the night, we do intermediate molecular typing. We use sequence-specific primers that can amplify specifically HLA-A1 or HLA-A2. Another method that we use are primers that amplify all the alleles of a particular locus, so it amplifies all HLA-A alleles, and then we come in with sequence-specific probes to determine is the amplicon HLA-A1 or HLA-A2. We always talk about HLA as being very diverse and polymorphic. However, there's a great deal of homology within HLA because it evolved through gene duplication and gene inversion. And so it's this combination of high polymorphism and high homology that causes HLA typing to be a high complexity field. And so in this diagram, I'm showing you different HLA A alleles. The patient is either an O201, O202, or possibly an O203, O206. And using Sanger-based methods, we know that the patient contains the red polymorphism, the green polymorphism, and the blue polymorphism, but with Sanger sequencing, we were unable to determine, because we sequence all the molecules together in one shot, we don't know whether these polymorphisms lie in cis, encoding O2O3, or whether these polymorphisms lie in trans. And this kept us from really being able to do high-resolution allele-level typing for our bone marrow colleagues. However, with the advent of next-generation sequencing, in which we're sequencing one molecule at a time, we can use bioinformatics approach to find specific sequencing reads that link the red polymorphism to the green, and a read that will link the green to the blue. And from this type of single molecule sequencing, we can now achieve very specific allele level typing. This is the nomenclature. Let me see if yeah, good. This is the nomenclature for HLA. For your purposes, it's not that bad. It's really not that bad. And so I've been told it's like alphabet soup. But really, you just look at after the dash, and that will tell you what locus is you're looking at, HLA-A. The first field is the allele group, so HLA-O2. And the second field is the actual allele encoding a unique protein. So in this case, it's O101. Everything beyond that has to do with crazy immunogenesis. I have to tell you, the third field is coming to a transplant report near you, because it's this third field that will tell us the level of expression. So different alleles can have different levels of expression, and so that is coming, but not in the near future. These are the classical HLA molecules that we type for transplant. Class 1 molecules consist of a single polymorphic chain, ABC, and class 2 molecules are comprised of alpha-beta heterodimers, and both the alpha and the beta chains are polymorphic for DP and DQ. For DR, the alpha chain is non-polymorphic, and so we do not type for that. So again, in solid organ transplantation, we traditionally report the antigen group, so HLA-A2. However, for our bone marrow transplant clinicians, we report alleles, and this again shows you that you can be an A0201 all the way up to an O2-877. Another thing you'll notice on the reports is there are certain genes to which there's no typing information, and that's because not all of us carry all the same genes. And so to give you an example, I inherited a DR8 from my father, so I have a DRB1 and a DR non-polymorphic alpha gene. And then I inherited a DR12 from my mother, and the DR12 haplotype also carries a DRB3 gene. So I will have typings for 2 DRB1 genes, a DRB3 gene. Whereas if you are a DR8 and a DR1, you would not have any of these additional DR genes. So because HLA genes are nestled very close together on chromosome 6, they're inherited in blocks or inherited in haplotypes. And so HLA people are able to look at typing and immediately recognize things that are not in linkage disequilibrium and look strange. So there was a revolution with all of this polymorphism. Testing for HLA-specific antibodies was difficult because it was difficult to test so many different analytes, so many different proteins. And so Luminex testing was really revolutionary for the HLA laboratory. And you may think Luminex testing was so revolutionary because it was so sensitive, but actually it was revolutionary because it allowed us to multiplex. So each of the Luminex beads, we can test 100 Luminex beads in one test tube. Each bead is infused with two different proportions of 2 red dyes so that we can identify each bead individually by their specific fluorescence. And then we can coat each one of these beads with a different HLA molecule. And then we can simultaneously test 100 different HLA molecules with 1 drop of serum. And we are advancing, and the laboratories are getting new machines into the laboratory, so soon we'll be able to test 100 different beads simultaneously. There are different types of HLA beads, Luminex beads. There's our screening beads, which are low cost, and it'll tell you if the patient has antibodies or not, so it's HLA sensitization positive or negative. And we can determine some increase and decrease, but we don't know specifically what antigens are increasing and decreasing. Next, there are phenotype panels. So these are beads in which it's coated with an entire class 1 or class 2 haplotype. So they strip class 1 antigens off of a cell and coat them onto the bead. So those are made in human cells, and they have the correct glycosylation and phosphorylation, the post-transplant modifications. And they're also in at a density that's more physiologically relevant. And then we have the famous single antigen beads. These are beads coated with one single allele per bead, and these HLA molecules are made recombinantly. So this is the assay. Many of you, I hope all of you, have visited your HLA laboratory. You take your HLA-coated beads, you add patient sera. Washing is critical to wash away nonspecific binding. However, it could also wash away low-affinity antibodies. And then we come in with an anti-IgG fluorescent antibody, run it through a laser, and the amount of fluorescent will tell us how much antibody is bound to a specific bead with a certain HLA antigen on the surface. And the amount of antibody bound is relatively proportional to the MFI, or the median fluorescence intensity. But this is a semi-quantitative test because HLA antibodies are polymorphic, and it's really, when you have an A2 antibody, you have multiple antibodies of different affinities binding to slightly different epitopes. So there is interlaboratory variance in these tests because they are so sensitive. There are differences in washing techniques. We all are using different bead lots that add variability. We also now have, we're using different Luminex machines in the different laboratories, and so even the laser age can differ in the intensity of the signal that comes out. And this was a study done by Elaine Reed in which they exchanged 20 serum across 7 laboratories, and the laboratories had complete consensus on what antibodies were present in that serum, but everyone submitted their raw data, and what they showed was with antibodies at 3,000 to 10,000 or 10,000 to 25,000, we got pretty good reproducibility in the MFIs with an acceptable percent CV. However, when you go below 3,000, particularly in the very low end of detection, there was an extremely large coefficient of variance because of these subtle differences in bead lots, washing techniques, and the Luminex machines that we use to read. So within your laboratory, you should have good reproducibility, but when you read papers and you look at a cutoff from some other laboratory, it may differ from your laboratory because of these interlaboratory variances. We published a paper very early on in 2009 from Johns Hopkins that certain sera, especially high PRA sera, contain interfering factors. And beautiful studies went on to show that IgM can bind the Luminex beads and bind the HLA and inhibit our detection of IgG binding. So the pink antibodies here are HLA-specific IgG from patient sera. We also know that antibodies that are good in complement fixing can actually fix complement that's present in the serum. And then when we come in with our detection antibody, this complement can cause interference. And thus, instead of reading that there are 5 antibodies bound to this bead, we're only reading that there are 2. So there's a significant underestimation in some sera. Treatment of the sera with EDTA or heat or DTT can get rid of IgM and complement. At Johns Hopkins, we used a more robust method, and we would pass patient sera over a Mellon column, which actually enriches IgG molecules and gets rid of complement, gets rid of IgM. And we found that this was the most robust way to get rid of all of serum interference. PRA. PRA is actually a very old terminology, and it is a term that the laboratories use to communicate with the clinician of the likelihood of finding a compatible donor within the donor pool. So in this beautiful slide stolen from the Canadian Blood Services website, I show a patient who's sensitized to HLA-A2. This patient has 1 antibody specificity, and he has a 46% CPRA, which means that out of a panel of 100 donors, he will be incompatible with 46, because 46 of those 100 will have A2 within their phenotype. So that is panel reactive antibody. What can CPRAs look like? You can have an 82% CPRA with just 2 antibodies, an antibody to A2 and an antibody to DR52, because those antigens are so common in the deceased donor pool. You can also have a CPRA of 82 with many, many, many antibodies to less common antigens. And so CPRA is, again, a tool for looking at the likelihood of finding a compatible donor. CPRA does not define strength. So here I show candidate John Doe, who is listed at 2 centers. This is becoming much more common that patients are multi-listed. At center 1, his CPRA is reported to UNIS at 82%, and that's because they are listing A2 with a very low MFI of 1,000, and they're listing DR52 with a 500 MFI threshold. Center 2 has proven that they've been successful in crossing antibodies that are flow negative, perhaps at University of Colorado, for instance. And so they may not list these antibodies as avoids. And so that same exact patient with the same test results may be listed as a 0 PRA at that center. Here's another example how a CPRA does not define strength. You have John Doe and Jane Doe. Both have CPRAs of 82. However, John Doe is the same patient with these very low-level MFI antibodies. Jane has extremely high-level HLA antibodies. And so our CPRA tells us the breadth of sensitization, but not the strength of the antibodies. So how do these differ? The percent CPRA tells us the incompatibilities within the entire donor pool, the likelihood that we're going to find a compatible match, and the DSA tells us, is there an incompatibility with a particular donor and the HLA mismatches within that donor's phenotype. And it's these tools that we perform virtual cross matches. So virtual cross matches is when we take retrospective testing data in the HLA lab and we produce a risk assessment, and we talk about incompatibilities. So in a virtual cross match, we examine the HLA donor typing, which at this point is an intermediate typing. We know what antigen groups, but we do not know the allele, because to do next generation sequencing, it takes 3 days currently. We do not have 3 days to wait. We have 3 hours. And so we're using intermediate HLA typing. We're looking at the patient's HLA sensitization, both current and historic, to determine is their DSA present or absent. And if it's present, whether it is at a level that it incurs great risk. And so some centers cross DSA in the middle of the night, and so it's up to the HLA lab to provide a risk assessment of what is the risk of crossing that HLA antibody. Oh, and I forgot to say, virtual cross-matching is not new. This is a paper in 1964 published by Paul Terasaki, in which they used serological panels to identify the CPRA of a patient and what the patient had antibody towards, and they used serology to type the deceased donors, and they looked at these incompatibilities and performed virtual cross matches as well. So here is a scenario. I didn't put this in a polling question, but you have a transplant patient who types as a DRB104 homozygote. A potential living donor is also typed at low resolution as a DR4 homozygote. The patient is sensitized, has class 2 antibodies, and the previous transplant was a DR1. Is this a great donor? Do you need a virtual cross match, or can you go directly to the OR? I think you need a virtual cross match, because when we actually do our typing to the second field, which we can get second field typing for most common antigens across the US. We know that the patient is an 0401 allele and the current donor is an 0402 allele, and DSA is present because, as I mentioned, there's a lot of sharing of epitopes between HLA alleles. At position 71, the patient's DR4 has a lysine. However, in that same antigen group, the donor is a DR0402, which carries this epitope or eplet at position 71, and that same eplet is shared with the previous rejected transplant, 0103. And so antigen groups help us tremendously. The most immunogenic epitopes are shared within an antigen group, but there are very immunogenic epitopes that lie between alleles within the same antigen group. So again, you can't assume by low resolution typing that there is no DSA present. And so HLA mismatch is seen within the context of self. And to explain this, this is a space-filling model of an HLA class 1 molecule, HLA B51. And when you compare it to a consensus sequence, these are all the polymorphic residues on B51. However, if I'm a patient and this is my type, I have some of these polymorphic residues within my own HLA phenotype, and therefore I only see 2 of these residues as foreign. And looking at another individual who has a B35 in their own phenotype, B35 is highly related to B51. And so if I'm a B35 transplanted with a B51 mismatch, I only see 1 residue as foreign. And so the degree of differentness depends on both the patient's typing and the donor's typing. Which brings us to eplet versus epitope. So an eplet is a made-up word by René Duquesnoy, brilliant man, in which he looked at amino acid triplet mismatches, both linear triplets and 3 amino acids that come together in conformational folding. And he identified these eplets that were confirmed by serology. So we knew antibodies were binding to this specific polymorphism, like the sequence alignment that I just showed you with the DR0402. But really, an epitope is the entire area to which an antibody binds. And these things are actually 2 different things, and they should not be confused with one another. So to give you an example of why people in the HLA field want to make that distinction is because this is a modified slide from one that Leo showed yesterday. So we have an antibody binding an epitope, and that epitope is comprised of 2 eplets, so 2 regions of polymorphism shown here in red. However, on a different allele, if we change the amino acid between those 2 eplets, so we change it from a lysine to a large arginine here, we disrupt the epitope. The antibody can no longer bind. Even though the eplets are still present, the epitope is broken. And so we know in the laboratory that sometimes, even though there's a clear eplet, the surrounding amino acid sequence can greatly impact whether an antibody can bind. And this has been shown beautifully by Kosmaliopsis in Craig Taylor's lab when he was looking at electrostatic potential and the differences even between the BW6 motif. So an eplet is not an epitope. That being said, why do we look at eplet? Because we have this fantastic tool called HLA Matchmaker that runs through the HLA sequences and counts eplets. And that software has made it very nice for us to look at a molecular mismatch, not an antigen mismatch, but a molecular mismatch. And this is, again, work by Kosmaliopsis, in which they just looked at the number of amino acid differences between the recipient and donors and found that as the number of amino acid differences increased, or the molecular HLA mismatch increased, the prevalence of de novo DSA increased. This is a paper from Catherine Tinkham's group out of Toronto. We know that different eplets or different amino acid mismatches carry different immunogenicities, and such that certain mismatches are more likely to ignite a de novo DSA than other mismatches. And here she looked that within heart and lung transplant recipients, being mismatched for the DQA gene, and particularly if it's a repeated mismatch to the DQA gene, you are much more likely to elicit de novo DSA. So there will be an 18th International Workshop in 2021 in in Amsterdam, in which we're going to dive into HLA epitopes. We need to define which are the most immunogenic epitopes, and so that we can better provide greater risk assessments at time of transplant. It is unlikely that we're going to match our recipient and donors at the HLA allele level. However, if we can imagine matching for the most immunogenic epitopes, and if we can imagine at least letting the clinicians know how much of a mismatch there is, and you can use it in personalized immunosuppression, I think we can do better than we're currently doing. So defining the risk for de novo DSA, cumulative HLA mismatch, not at the antigen level, but at the molecular level. We can use multiple different tools, and the workshop will be testing these tools against one another. We know that patients who are under immunosuppressed or have repeated infections and acute rejection all pose greater risks for de novo DSA. So the role of the HLA laboratory is to determine if HLA is present or absent, determine whether it's risky and it should be monitored, determine if it's injurious and it should be treated. So I'm going to give a brief example of some single antigen bead data. So this is a real case study. This is a man who presented for a heart transplant. He had received an LVAD and had received transfusions 2 years prior. The MFIs on this single antigen bead test are very low. They go from 1,000 to 300. However, this is a 100 bead set that we're testing here, and the top 6 beads are all DQ6. And for the HLA people in the audience, if there are any, immediately following it are DQ5, which is closely related to DQ6. And so one of the questions I get asked most often as an HLA director is, what is your MFI cutoff? What's your cutoff for positivity? And I always say, I have no cutoff. You know, I look at pattern. Our laboratory looks at pattern, because these 6 beads did not bubble to the top of a 100 bead panel by accident. And so when this patient was reported, we said that there was evidence that the patient had previously been exposed to DQ6, and DQ6 was a mismatch in this heart allograft. This alerted the clinicians to do some post-transplant monitoring, and on day 5, you can say, well, how do you know if that's real? It's so low. Well, on day 5, we had a significant jump, and now the MFIs are saturated at over 25,000. The DQ6 at the top, and very strongly beneath them are DQ5s, which share a lot of epitopes with DQ6. And so we really can do a lot of interrogation with pattern analysis that may be better than reporting MFI. And here's another example on the other extreme. This is a patient who presented, and they brought forth a live donor who was a B4402. When we did the testing, the top bead was B4402 at over 11,000. However, there was a high negative control bead, something that I don't think is often reported in the electronic medical record. And in addition, there was no logical pattern. B45 is related to B44, but the other B4403 bead was completely on the bottom. So this looked odd, and this is what we call a high background serum. We tested it on a phenotype panel. Now, the top bead reaction is only 500 MFI. The negative control bead is much better on the phenotype panel. And we see on this negative interpretation that all B44s and B45 beads are completely negative. So we reported this as a negative antibody patient. And all of that reactivity on the single antigen bead was false negative. And so I've showed you where I've called and reported a DSA at 300 MFI, and I've completely ignored an MFI of 11,000. And so the importance of interpretation and using what we know about epitope sharing. Is it injurious or risky? We know that MFI does matter, that the higher the level of antibody, the more likely you're going to get 6 IgG molecules within close proximity, which is necessary for activating complement. We know that patients make polyclonal HLA antibodies, and it's made of an array of different IgG subclass compositions. However, in all cases that I've examined thus far, IgG1 is by far the most common subclass within DSA. We have new tests that test for function. These are the single C1q or C3d assays, where we don't have a detection antibody specific for IgG. We have a detection antibody that detects the binding of C1q or C3d. These are less sensitive, but more specific, and we can tell a little bit more about the quantity of antibody. However, there are drawbacks to the functional tests. For instance, this is not specific for IgG. If you have IgM in your serum specific for HLA, IgM activates complement a lot more effectively than IgG. So remember that. It's a functional assay, but it's not telling you the amount of IgG DSA. This is work from Alexander Lupi that showed that certainly C1q positive DSA, which are higher titer and complement fixing, were associated with more severe antibody-mediated injury, as shown in glomerulitis and peritubular capillaritis and C4d positive staining. Very quickly, because I have 12 seconds. There are different expression levels of the HLA genes. HLA-C is expressed at lower level, and as are DR and DQ on the cell surface. And so we have to remember that you can have a low titer DSA with good HLA expression on the vascular endothelium, and you may not activate complement. You can also have a very high titer HLA-C antibody that also, because of low antigen expression, cannot activate complement. However, HLA expression within the allograft is extremely dynamic, and there are not enough good studies looking at what kind of dynamic with BK viremia, with acute rejection, how much HLA expression ebbs and flows. And certainly you can go with a high titer HLA-C DSA. The patient is completely non-symptomatic with no histological evidence of injury in a quiescent state. However, when you ramp up expression, that can change suddenly. And I would like to thank my teams, both in my research and clinical immunology lab at Duke, and I'd take any questions.
That was excellent. So one of the questions that came up, there was a number of questions. You did end up answering a number of them as they came through. The C1q assay, the C, using HLA-C in terms of calculating PRA or having its immunogenicity actually be maybe reduced because of tissue expression. Some of the other questions were the intra-lab variability that we see. Can we ever truly generalize one lab's experience or one center's experience to others and really correlate data across experiences across centers?
I think we can, but I think we're so fixated on the MFI. I think certainly at Hopkins, we really reported DSA kind of in 3 categories based on clinical outcome experience. We reported DSA positive by Luminex beads. We reported DSA positive sufficient to yield a positive flow cross-match. And we report DSA at the level of CDC positive. The CDC positive level DSA, that means you should treat today. The flow positivity meant at Hopkins that we would treat that DSA, and then a Luminex positive is up to the clinician to put it into context. Do we have an accompanied biopsy that shows no inflammation? Do we have dysfunction? Does the patient have BK viremia? And so we reported kind of at 3 levels that then triggered different clinical action. I think that is a much more reasonable way to report DSAs. And I think when Dori Segev first went out to look at HLA incompatibilities across centers, he asked me, Annette, what should be our MFI threshold? And I told him, throw MFI in the trash and collect cross-match data. And so that's what he did. In the studies by Babak Orande, they looked at Luminex positive, flow cross-match positive, and CDC positive, and certainly they saw very different clinical outcomes. And so I think we need to back up and take a more functional look at DSA levels.
Perfect. No, that's excellent. We're going to thank you very much. That was an excellent talk. So our next speaker is Mike Mengel, our pathologist straight from Banff, and now here, actually we have a number of Banff travelers, and we'll hear about what antibody looks like in the graft. Thank you.
Okay. So, my disclosures. First, we start with a question. After everything you have heard so far, do you believe in antibody-mediated rejection? Yes, it's a major cause of late allograft failure. No, it's made up by pathologists. It's the largest collusion in transplantation medicine. And it exists, but it's clinically irrelevant. All cases are mixed rejection anyway, so who cares? Sorry? Huh? It works? Are you sure? Standing. Okay. Wow. So when we started this fellowship symposium, it was different years ago. But so we come a long way. Everybody's aware of antibody-mediated rejection. Another audience question. The Banff classification for antibody-mediated rejection, you never heard about it? It's not used at your center. It's used by the pathologist but ignored by clinicians. And when you take that answer, please identify yourself. It's too complicated and only out there to make pathologists feel important. It's critical to guide care in the individual patient. So there is no right answer, but there's a lot of wrong answers. I didn't do that. Why did I? It did not advance. But anyway, can we see the question? No? Why not? Huh? I can't read sign language. The person like it didn't register. So that's too disappointing. Okay, so raise your hand. It's there? I do not see anything. Anyway, so who is aware of the Banff classification? Wow. Are you using it? Oh. Okay, so you heard a lot about what antibody can do, and it can present clinically with a different course of the patient, but it's important to understand that it's actually one disease process and one immunological mechanism, as Dr. Fairchild explained to you, but it can present due to various factors, but mainly due to what Dr. Jackson told you, that the characteristics of antibodies and the combination of different antibodies can cause a very different phenotype. So antibody can be a very hyperacute rejection phenomenon. That fortunately doesn't exist anymore because we do cross-match. But when you transplant an organ without doing cross-match, so don't do it, right? Because you can immediately lose the graft. But then there is people who have been sensitized and went either through desensitization or the antibody production silenced, but still they have memory. And then you transplant them and they get re-exposed and they have a very early memory response or pre-existing antibodies and get early antibody-mediated rejection. And then there is patients who develop antibody late after transplantation. So these main phenotypes can cause a different clinical picture you deal with, but still it's antibody-mediated injury. And we tried to reflect that in the histopathology phenotypes, and therefore there are terms like active antibody-mediated rejection, chronic active antibody-mediated rejection, and also frequently people use chronic antibody-mediated rejection in a certain setting. So it was introduced into the Banff classification in 2001 and then published in 2003, but the Banff classification existed since 1991. And it's not that antibody-mediated rejection started to exist in 2003, okay? It was there before, but it was not appropriately diagnosable, or there was no consensus. And there was also a belief that it was an epiphenomenon of the T-cell rejection, because T-cells, as you heard in all the immunology lectures, are needed to give help for antibodies to be, or B cells to mature to plasma cells and then produce antibody. But the first iteration of the classification of histological criteria for diagnosing antibody-mediated rejection was introduced then with this seminal paper. It's one of the most cited papers in the transplant literature. And since then, because suddenly when you can diagnose something, look, it's there, and it actually is relevant. And it was identified as the major cause of allograft injury and failure, and presenting a major challenge in clinical management. So antibodies, so this is after the talk, so Dr. Fairchild, all the immunology, right, way above my head. And then Dr. Jackson comes and says, don't worry, there's just a spreadsheet with 800 alleles, and we know it all, right? So this is how antibody-mediated rejection works for me. There is an antibody, it binds to the endothelium, and it does no good. Okay, that's antibody-mediated rejection, and there's three main pathways that you heard: complement activation, antibody-dependent cellular cytotoxicity through the Fc receptors in K cells, neutrophils, macrophages, and mainly also the direct injury, which Dr. Fairchild explained in detail very elegantly. So keep these mechanisms in mind, because where in histopathology or in diagnostics, we then look for the morphological correlates or the molecular correlates of these mechanisms to achieve diagnostic specificity. And the first breakthrough in all this was the understanding that when you activate complement as a main pathway of antibody-mediated injury, during the complement cascade, there is this one step where you split C4 into C4a, B, and D. Why is there no C4C? Oh, never. Nobody knows. Does anybody know why there is no C4C? No. So usually complement components are either degraded quickly or washed away with the blood flow. But the uniqueness of C4D is it is covalently bound at the site where this step of the complement, the classical complement cascade, happens. So this allows us to use antibodies against C4D and to detect it in the tissue, and that's the famous C4D stain. And all what it tells us is actually that at this staining site, complement got activated. The function of C4D is only superficially understood, but detecting it says, okay, complement through the classical pathway was activated here in this site, and there is detailed electron microscopy, gold labeling stains showing that it's actually on the outer side of the endothelium. So you see it in kidney as a circular stain, you can see it in liver, you can do it by immunofluorescence, you have it in heart, you have it in lung, you have it in pancreas. That's a common denominator. When donor-specific antibody binds on the transplants and activates complement, we can detect it most of the time. And there is a scoring schema in Banff, okay? C4D 0, 1, 2, 3, just essentially says how extensive the stain is, how many capillaries show a linear circumferential stain, and you can use both the techniques, and they have slightly different sensitivity. And then the second major finding in the biopsy is, as Dr. Fairchild explained, the pro-inflammatory effect of antibody interacting with the epitope attracts inflammatory cells in the microcirculation. Again, peritubular capillaries, this is an endothelial cell, these are the inflammatory cells. The same thing in the heart, endothelial cells, inflammatory cells. That's a myocyte. That's a tubular epithelial cell. This is lung. Lung is really, that's why I'm not a lung pathologist, right? Where's the capillary? I could show you whatever. This could also be I don't know, a high-resolution image of an advertisement of a car sales unit. There probably is a capillary. But the fact is, usually lung is so delicate and without cells. And when you have a very busy lung tissue, it's inflamed. Same in the liver, there's a bile duct, and here is the periductular capillary. Same in the pancreas, islet cells, but here is the capillary. And you can stain, these are neutrophils and macrophages. So usually your small microcirculation is not loaded and stuffed with inflammatory cells. And that triggers the cascade Dr. Fairchild says is these cells can cause the release of coagulation factors, activate further pro-inflammatory chemokines, the endothelial cells, and essentially disrupt your microcirculation. That's not good in organs like the kidney or the heart, which are highly dependent in their function from a functional microcirculation for all the oxygen exchange. And in the kidneys, the uniqueness that you also have the same microcirculation, the glomeruli, which all of you kind of need on a daily basis. And when they are highly inflamed, it reduces glomerular function. And then, when this pro-inflammatory environment persists, the endothelial cells, this is electron microscopy, that's an erythrocytes, and that's a swollen activated endothelial cell, undergo injury, apoptosis, and then your microcirculation starts to remodel, because when they lose their functional endothelial surface, they try to repair, and that is first by remodeling or reproducing base membrane, and you see multiple layers of this. It gets thicker. Usually it's just one layer is your basement membrane around a capillary, and that facilitates the exchange of all the metabolites and oxygen. And when this suddenly is 10 times thicker, you have difficulties in that. And the same happens in the glomerulus. You have these typical double contours and thickening of the basement membranes. You have inflammation still ongoing. And then you can see by electron microscopy, these are endothelial cells, podocyte, 2 layers of newly formed basement membrane, and there is everything not useful interposition between these dead cells, cell material, fibrin, whatever. And that hinders the transport across this delicate system of your glomerular basement membrane. So, conceptually, the diagnosis of antibody-mediated rejection. You should have antibody, and how nice would it be if there would only be one antibody, right? And not 8,000 million. And then evidence that antibody acts on the graft, because there's a lot of patients who have donor-specific antibodies, but they do not act on the graft. So just detecting it in the serum is not good enough. You need to know whether it actually does something in the graft. And that's a bit of a complicated part, how effective what antibody is in an individual patient. So we look for this evidence either by complement deposition or the pathology with the microcirculation inflammation, and then in the chronic setting, whether you have tissue adapting to this ongoing injury by duplication and remodeling. Okay? And remember, I'm constantly misrepresenting facts to you in a certain light. All these things, except C4D, this pathology, you can see in other diseases. It's not absolutely specific for the diagnosis of antibody-mediated rejection, none of the organs. And that then, together with the million different antibodies, unknown properties, it's a bit of a challenge to figure that out in the individual patient then. But C4D is very, very, very specific when it's positive. So we have these features described for the pathology of the different settings of antibody in most organs, but remember there is still no consensus criteria for antibody-mediated rejection, for example, for the chronic state in heart and lung. Bowel there, just way out there. Composite tissue has also still a challenge in defining antibody-mediated rejection in the chronic setting. And again, when you do not have diagnostic criteria, right, how would you know what the prevalence of the disease is when you're not diagnosing it? Oh, yeah, it's 0, so it's not a problem in that. You just don't have criteria. So, there's a lot of focus on these endothelial changes, and they can be very early and very subtle, and especially in sensitized patients. And you can see them by electron microscopy. This endothelial swelling I described, and also in heart and kidney. But again, also when you're hypertensive or when you have other stress on the endothelium, you see these changes. They are not specific. The Banff classification has a grading system for that, which confuses people, but we have criteria to say when you have minimal capillaritis. That's for the interested reader. There's recommendation when you should do electron microscopy, and I think it's important for you to know that when you look at a pathology report and you have a pre-sensitized patient, for example, and it's very early, that you read the electron microscopy section and look for these features. But usually a pathologist would hint towards that in the diagnostic comment. And again, this famous Banff table that we put everything in 0 to 3, scoring criteria, and there's challenges with reproducibility, but at least it's a starting point for the consensus around that. So what you need to understand, antibody-mediated injury, when it charges your graft, so assume you have a DSA and the DSA is acting on the graft, this is a time-dependent evolution of what the pathology is and the clinical development. And it depends on when your patient shows up in the clinic and when you do a biopsy, what you see in the biopsy of the same disease, right? So people get confused saying, oh, I have chronic active antibody-mediated rejection, and this person has acute antibody-mediated rejection. These are not different diseases. It's like saying you have early stage lung cancer and advanced stage lung cancer. Still lung cancer. And here's the question. After all this, what is the major histological correlate of active antibody-mediated tissue injury? The learning success of this event. Okay, who said tubulitis? So it's an interesting question. So both C4D and microcirculation inflammation are features of activity, right? Antibodies acting on the graft. And it was a little bit unfair because I didn't show you the data yet around how reliable C4D is. But both of those are correct. No, not double contours. That's chronic. Okay, so there was a big C4D hype. When I was a fellow, C4D antibodies came out, and we thought, we got this, right? We stain, and then when C4D is positive, it's a super specific marker. Just yes, no, that's antibody-mediated rejection. Bang. And then people did research, and it turns out, and I like this study here done, done in Minneapolis. So when you work in pathology, one thing you should never do is go back to your old reports and double check whether they are right, okay? Never do that. So they went back before they started C4D staining and then stained everything again and found out how many cases they signed out wrong, right? So, but what they found is that actually, because they had to follow up, what actually presented as antibody-mediated rejection, they found the antibody and had microcirculation. Half of the cases are C4D negative, and they have the same outcome when they are C4D negative but have microcirculation inflammation. So then others found that as well. There's various studies. So C4D has limited sensitivity. It probably only picks up 50% at best of all antibody-mediated rejection cases. So why is that? Of course, one thing is your assay sucks, right? You're having that all false negatives. There's limited sensitivity of the assay. And then the second is, as Dr. Fairchild elegantly showed, we have at least 2 different pathways of antibody injuring the graft without fixing complement, right? That's a major source of it. So alternative diagnostic approaches, landmark paper by Luis Hidalgo looking at patients with DSA and without and the gene expression in the graft and showing that when you have DSA and antibody-mediated rejection, the major transcripts you pick up in an unsupervised study are endothelial transcript and K-cell transcripts and some inflammatory transcripts. And now that we understand the mechanisms of antibody-mediated injury, and after everything Dr. Fairchild showed you, that makes sense, right? And with that, they carry some specificity as molecular markers for antibody-mediated injury. And we could also show that even at the electron microscopy level, that the activation of these genes correlates with endothelial swelling at the ultramicrotome level. There are now assays out there you can buy, or you can send your biopsies there to get these genes assessed, or you can do it in-house. So there is a gene panel you can run on the formalin-fixed paraffin-embedded biopsies at home in your basement and get this gene expression. So it's not just research, it's come into the clinic to do these tests as an alternative to C4D. So here's where it starts getting complicated, that the pathology lesions, which we thought are all associated with either antibody-mediated rejection, yesterday we talked about T-cell-mediated rejection, can actually completely overlap. And they can not only overlap because they are not specific, no, because patients can have mixed rejection. And the further you're out, or especially when you're non-compliant, mixed rejection is a common clinical phenotype. And that's where really then you look at a biopsy and say, oh, maybe this, maybe that, and that's where people really get angry. And this is a landmark paper published in Lancet by the Paris group, essentially doing what is called here, it's a principal component analysis and finding out you can have pure T-cell mediated rejection, you can have pure antibody mediated rejection, and you can have T-cell mediated rejection phenotypes with vascular involvement and antibody mediated rejection phenotypes with vascular involvement, so arteritis, and they overlap. So certain lesions are more prone to be in mixed rejection, and some have some more specificity. As a rule of thumb, when you see glomerulitis and you have a DSA, you have an antibody-mediated rejection problem, and something else potentially. And the other thing which was then contributing to this complexity is that rapidly progressive vasculopathy, so arteriosclerosis, which is a common finding in kidney transplant because all your patients are hypertensive or they have other things going on, is actually in patients who have DSA more accelerated and faster progressing than in the regular hypertensive patient. And that's not only in the heart, where it was kind of known in association, but also in the kidney. And the more we get the antibody problem somewhat under control by treating the patients to some extent semi-effectively, you will see more of these late chronic lesions again. Okay, so here is everything I told you to this point, then translated into the Banff classification. And the major change to the Banff classification from 2003 was in 2013 to define criteria for a C4D negative antibody-mediated rejection, because between 2003 and 2013, it depended on the positivity of C4D. But then we found out, okay, 50% of false negative or are negative, and then you just miss 50% of the cases. You need 3 criteria, as I said. Oh my God. Histologic evidence of acute tissue injury, including one or more of the following. So you can have the microvascular inflammation, thrombotic microangiopathy, intramural arteritis, or acute tubular injury. That's a rare phenotype but can exist. Then the second one, interaction of antibody with the endothelium, ideally C4D, but if that is not there, moderate microvascular inflammation. So you see this criterion is in both. So when you have G plus PTC 2 or greater and a DSA, listen up. Yeah. And we introduced molecular markers back in 2013, and there were none. But we said, just in case, before we revise the classification again, we put it here. And then the third is, well, in 2013, you should have an antibody when you diagnose antibody-mediated rejection. Kind of logic, but not always, because as we heard, there is a lot of cases having the pathology, but a NET can't detect the damn antibody, right? And then you say, well, HLA lab sucks, right? Okay, chronic is the same approach, but you want to see these chronic changes with transplant glomerulopathy or peritubular capillary basement membrane multilayering. And that frequently you have a combination of these chronic changes and microvascular inflammation. So that's where the term chronic active comes from. Because there are rare cases where you only have double contours but no microcirculation inflammation. Those patients actually do better. They do not seem to progress. So the goal of the field is to control microcirculation inflammation. And to this point, we have not been very successful in that. So that is just the reference. Read that paper. And then people went back and said, did these changes make sense to include C4D negative antibody-mediated rejection? And actually, the rate of diagnosing ABMR doubled from 18 to 36% in this study. And the patients then included had the same outcome than the C4D positive. So it was a reassurance that the changes in the Banff classification were accurate, and actually set the stage now to do clinical studies, because we can enroll, per definition, C4D negative ABMR. Because before that, they all had to be C4D positive, and then your control arm probably had all the C4D negative ABMRs, and all trials failed. And again, I cannot stress this point more and more, because it depends on when you do the biopsy in this process. When you have a patient who was not in your clinic for 2 years for whatever reason, and then they come back and suddenly have proteinuria and the creatinine is somewhere and the GFR is down, they didn't contract ABMR yesterday, right? They were running around with that for years. And when you then do the biopsy late, you see a different pathology of the same disease than doing the biopsy 18 months before. So take that into consideration, that these are not different entities. So in 2017, an amendment was made that, because we had some challenges where people said look, I have the pathology microcirculation inflammation, C4D is positive, I do not have the DSA yet because they take the Friday off and the result is coming later, right? And can I not call it antibody-mediated rejection? So there is now a criterion saying when you have sufficient microcirculation inflammation and C4D positivity, you can call it antibody-mediated rejection even without having a DSA. And it becomes more relevant because sometimes really there is no DSA with the current tools we have to detect. Or there is the alternative that it is a non-HLA DSA, which most labs do not routinely test for. But you have the full-blown pathology and you need to do something for the patient. So that was introduced. And there was then a study done in Belgium by Martin Naessens, and he presented that last week at the Banff meeting, how many patients he actually has with the antibody-mediated rejection pathology, but no DSA. And there were, in his cohort of 1,000 patients, 123. And some were very early, and it probably represented ischemia reperfusion injury, but some were later, and it was probably antibody-mediated rejection without detecting a DSA at that stage. But he could show that the outcome of those with DSA and antibody-mediated rejection pathology is worse than those without a detectable DSA in his cohort. And that, again, there is various reasons for that, and there is some heterogeneity in that group, because also what we do not understand is what role a certain B-cell memory plays in your antibody production. Some memory cells have a greater capability of producing DSA or at a later time. But nevertheless, there is a group in the Banff classification where you can be now morphologically ABMR with C4D and have no DSA, which we would call ABMR, but half of those patients are actually not behaving like ABMR. So there's still a cohort we do not really understand and probably misclassify or potentially. So diagnosis of antibody-mediated rejection is not just one thing. It's you need to understand your HLA report, which is super easy when you get these big sheets, right? And there's bar graphs and numbers, and I look at them and say, hey, whatever. And yeah, and I call Trish Campbell and say, hey, what does that mean? And then there is the different pathology. I'm not saying that the Banff classification is easier than your stuff. That's why we do meetings together. It's really nurture on each other, right? Who becomes more complicated. And now we have molecular tools, and it is really the clinical history is important, and to integrate that into a final diagnosis of a disease which goes in phases and where staging is also relevant for the prognosis of the patient. So here are all the key reference you need to read next week. And then, you believe, and we skip that question. Can we skip that question? Because we got the answer, kind of. So here again, can we do that? Well, we can't do that question, right? You got it now? Okay. So now I want to know whether that changed. We're 10 seconds over, so. And they promised me we can do pre and post. We can do pre and post. Wow. Well, yeah, my talk improved you by, wait a second, 12%. That's great. It's used by Alpha, but ignored by, look, the ignorers changed. I converted you. Okay, with that, I'm happy and I can go home. Thank you. Wow.
Okay, so two just quick questions. One is, given the fact that C4D is so spotty, and in particular in lung transplant, et cetera, how close are we to the validation of these molecular markers or transcripts as a diagnostic tool?
Yeah, so what in the development of these molecular markers, what has happened is that the analytical accuracy has been validated. You can do the technologies, they can be reproduced, there are centralized labs, there are technologies you can do in your lab. So we know what genes are most strongly associated with antibody-mediated injury. What we do not know is who to test when, and what actually to do with high expression of NK cell and endothelial transcripts, right? So the clinical utility has not been validated, and the most critical part is, does it improve patient care and outcome at the end of the day? For a randomized trial is missing, saying one arm is managed with using molecular tests and one arm is managed conventionally, and is there a difference in outcome? And I think there is a good chance that it could not be, right? Because we have no accurate treatment on the other hand. Yeah.
One last question, really quick, is you commented that these findings in isolation are nonspecific. For example, microvascular inflammation. Could you comment a little bit about what, if in the absence of an antibody, absence of other diagnoses, what comes to mind in a pathologist's diagnosis?
So peritubular capillaritis you can see in T-cell mediated rejection as well. And you can see it early post-transplantation in severe ischemic injury and acute kidney failure. And glomerulitis, you can see in any early stage recurring glomerulonephritis, like dense deposit disease, which recurs 100% in the graft, can present identically with glomerulitis at the beginning, or MPGN, or infectious glomerulonephritis. So you need electron microscopy to rule out deposits, for sure.
Perfect. Okay, thank you very much. Okay, so our final speaker for this session is Kristen Rogers. She'll be talking about what happens when we start seeing, and Betsy Vernon. Okay, so Betsy, you're coming up first? Okay. So we have a tag team for what happens when patients don't take their meds and we start to see antibodies. Thank you.
Okay. So we're going to talk about medication adherence, and we're going to start a little bit by talking about why it's important in a few case scenarios, but I think really importantly for you guys, we're going to talk about interventions to make it better, and so some tools to use in the clinic, some ways to actually understand adherence in your patients, because obviously if you don't ask, they're not going to tell you, and why it's so important. So the first slide here is really just to show you, this is kidney outcomes over time, the risk of rejection and the one-year survival of the graft. And you can see, and this has been shown, in all the different organ types, that we've had this tremendous improvement in our outcomes over time, which is largely attributed to advances in the medications that we use to protect these grafts immunologically. However, these are really complicated medication regimens, and I think we all need to really think about what it might be like for our patients to take this many pills a day. Many patients come in on a lot of pills, but certainly in the first few months after transplant, this can be really overwhelming. Not all of our patients fully understand all of this, despite all of the education that we do before the transplantation. These are medicines that not only are large in volume in terms of the number of pills that they need to take, but they have very complex narrow therapeutic windows, that require pharmacologic monitoring and have a lot of drug-drug interactions. And so this is actually really complicated for people, even when they're doing everything exactly as prescribed. And every person on the team really has a role to play in terms of communicating with patients about their medications and their adherence to it and their understanding. It includes all of the providers in the hospital. Also, a big part of the care team, I think, is figuring out to make sure that the patient's actually taking the medications as they're prescribed. And then obviously, I think we all need to acknowledge the critical role of the pharmacists on our team. You know, they're a required part of our team for the reason, and they have a lot of tools that can help us actually improve adherence in patients when patients are struggling. So we all know medication adherence is important. It's improving graft survival. We educate patients before the transplant. We're prescribing medications. They're surrounded by doctors and caregivers after transplant. So how is it possible that so many people are not adherent? And so these are just a few case examples for things that come up frequently that might sort of point you in a direction for the type of intervention that would help them. So this is an example of a 45-year-old woman who's 2 years post-transplant. She's very busy. She's a working mom. She's working full-time. She's trying to get back into the groove at work. She's traveling on the weekends to support her kids in her activities. And it's very difficult for her to come and get routine lab draws at the right time of day, and sometimes misses doses of medications just because her life and schedule is so complex. But she's tech savvy, and maybe there are some ways that we can support her with reminders to get these things done. What about education and comprehension? So this is a 68-year-old woman who's 3 months post-transplant who comes in with rejection. Somebody who was transplanted, a liver transplant in the hospital as a high MELD case, as a late referral, so we didn't have a lot of time to educate the patient. English is not her first language, nor is it the first language of her only caregiver, who's her sister, who lives with her. And now she's coming in with rejection, and when you really drill down on what she's been taking at home, you realize that there's been a bunch of medication errors. So how can we help this type of patient? What about all the side effects of these medications and toxicities? So this is a 74-year-old man who's 6 weeks post-liver transplant, had a prolonged post-operative course with lots of complications of immunosuppression, including prolonged delirium, terrible tremor. We had to change his calcineurin inhibitor. He came back in with C. diff colitis and has had sort of terrible GI complaints since then. And he now comes in, he's exhausted, he's fatigued, he has elevated liver tests, and he sort of owns up to the fact that he sometimes skips doses of these medicines because he feels terrible, both neurologically and also from his GI side effects. And finally, these medicines are expensive, and we spend a lot of time sort of authorizing all the medicines going into transplant, but insurance changes, co-pays are high. This is a 21-year-old who's 6 months post-kidney transplant. He's had a lot of complications, delayed graft function, multiple admissions for rejection. He's now coming back in with recurrent rejection, and we realize that he's been actually rationing his medicines because he can't afford the co-pays. And now he's in a worse scenario requiring thymoglobulin. So what is adherence? It's different than compliance. So compliance is sort of this passive act that the patient is going to follow your instructions and take their medications. But adherence is something more specific. It's not just that they report that they're taking their medicines, but it's that they're taking their medicines exactly as prescribed. If it's prescribed twice a day, they're taking it twice a day. And they start the medicine when you ask them to start, and they stop it when you ask them to stop the medications. And so adherence is really in phases. It's not sort of just one thing. It's about when you initiate the medicine, whether it's implemented exactly according to directions, and then whether it's either persistent or whether when you ask them to stop the medicine, they stop it. And so failure to follow those directions in each of these phases has negative consequences in terms of their graft outcomes. Medication non-adherence after transplant is really common. It's much more common, I think, than most clinicians think when they're in the clinic. And your patients are also non-adherent. You know, I think I can't imagine any of my patients not being totally following all the directions, but it's really true. And this has been shown over and over again in the literature. This is a meta-analysis that was published in 2007 that reported that transplant recipients have non-adherent rates that range from 28 to 52%, depending a little bit on the type of transplant and also your definition of non-adherence, and that it actually differs depending on the type of transplant they have. I'm proud to say that the liver group does the best. But, 35% of renal transplant recipients are non-adherent. This has to have a huge impact on their graft outcomes. And this is an interesting study where they actually looked at the evolution of non-adherence from the pre to the post-transplant period. This is a large prospective cohort, the Swiss transplant cohort study, where they actually asked questions about adherence at multiple pre-specified points before and after the transplant. And you can see here that obviously pre-transplant non-adherence is alarmingly high. Then they get transplanted, but over time, probably as your visits space out and they're feeling better and lots of other things that happen, non-adherence creeps up. And so this is something that's important to assess, especially in the late visits after the transplant. And when they looked at this sort of in multivariate analyses, both pre-transplant non-adherence and also the time from the transplant were the strongest predictors of whether the patient was non-adherent. Some other things came out here too in terms of patient age and other things, but this is really important. And I think all of us see this. We have patients that we list for transplant that we know are not really taking all of their medicines the right way, and these are the types of patients you really have to look out for after the transplant. So what is the cost of non-adherence? It's very, very significant. It's the third leading cause of graft loss and infection after infection and rejection. It's associated with decreased 5-year graft survival. This is specifically in kidney transplantation. It also is associated with a huge economic cost. And actually, patients that are not adherent to their medications, even though they might be spending less on medicines at that time because they're skipping doses or not taking medicines, in the long run, because of all the consequences of not taking their medications as prescribed, they're much more expensive. And these are just a few examples of studies that have shown this. So this is a very large study looking at USRDS and Medicare claims data, where they actually tried to put patients into different groups in terms of how non-compliant they were based on the prescription data, and showed that the more non-compliant you are, not just as a yes/no question, but the more non-adherent you are, you have progressive risk of graft loss and death after kidney transplantation. And in addition, there's a very significant increase in the cost associated with these patients. So patients with persistently low compliance level incurred $33,000 more in medical costs over 3 years compared to those with excellent compliance. So how does this actually work? How does non-adherence cause increased risk of graft injury? There's probably lots of mechanisms there, and you guys just heard a bunch of lectures about how that works, but a little bit of the evidence going for this. So this is another prospective cohort, large cohort, where they were able to look at de novo DSA and late graft loss in a group of kidney transplant recipients, and almost all of the patients that had de novo DSA were patients that had documented non-adherence in their medical records. And in fact, both de novo DSA and non-adherence were highly predictive of graft outcomes in this cohort. And then 90% of the patients with clinical de novo DSA had a history of medication non-adherence in their record. So this is a group of patients that we really need to target in order to make sure that we can prevent these outcomes. So how are we going to know whether our patients in clinic are non-adherent? The first thing is that you have to ask. We're all busy. We run through lots of other things going on with the patient at the time. Sometimes just reconciling their med list takes 10 minutes. But in addition to knowing what they're supposed to be taking, we have to know whether they're actually taking it. And I think that requires actually asking real questions about their schedule and how often they miss doses and these types of things. So there's a lot of different things that are available out there, tools that we can use to assess this. We think that the ones that are most common are just sort of objective interview, obviously, measuring their actual prograph and cyclosporin levels when we can. There's a bunch of standardized questionnaires that are available now. And also, obviously, intermittently talking to their pharmacy about whether they're actually refilling their medications. And, this is one sort of theoretical construct. We're looking at the coefficient of variation and people's tacrolimus levels after transplant is one way for us to do this. I think in this paper, and we have to sort of use a little bit of caution early after the transplant because there's so many different things that go into variability in these levels. But in stable patients out in that 3-year post period where they actually are at high risk of non-adherence, this might be something for us to start thinking about in terms of how we monitor. Okay, I'm going to turn it over.
So in the second half of our presentation, we're going to talk more about the identified risk factors for non-adherence as well as some of the interventions. So when you look at the data supporting risk factors for non-adherence, it's really separated into two types of studies, the qualitative studies and the quantitative studies. So the quantitative studies look at the empirical associations between punitive risk factors and adherence, whereas our qualitative data is focusing more on asking patients what factors really truly affect their adherence. In 2003, the World Health Organization developed the systems approach for studying adherence, and this really set the framework for a number of studies looking at risk factors for adherence after transplant. The most commonly studied specific factor, the social and economic factors in transplant, include the patients who have been identified are the patients who are typically younger, those who have poor social support, minority ethnicity, low social economic status, and health literacy. So these are patients that you may want to target interventions towards after transplant for adherence. Therapy-related factors that have been identified with non-adherence include complex medication regimens, so patients having to take medications 4 or 5 times a day, medication side effects, most commonly those that are more cosmetic in nature, lack of education about medications or understanding as to what they're taking their medications for, as well as a lack of a reminder system or consistent routine. Of the patient-related and psychosocial factors, as Dr. Vera has mentioned earlier, the history of non-adherence is probably one of the most significant factors that correlates with non-adherence post-transplant. In addition to non-adherence, psychosocial, psychological distress, substance abuse, interruption of routine, forgetfulness, as well as negative beliefs in medications and not really feeling that they're going to work for them, those things also contribute to patients' non-adherence. Looking at healthcare system-related factors, these include medication costs, which we'll talk a lot about in a little bit, poor access to care, poor aftercare planning, as well as poor provider and patient communication. And then finally, looking at the condition-related factors that have been associated with non-adherence, development of comorbidities, so these are areas that we can intervene post-transplant on side effects, time post-transplant, which was illustrated previously, so further out from transplant, patients have a tendency of being more non-adherent, and then poor perceived health. When looking at some of the qualitative data, this was a nice systematic review that looked at various factors or feelings that patients had about taking their medications that were associated with various levels of adherence. So in patients who were very adherent and had vigilant medication taking, they felt that they had to feel like they were protecting their new chance at life. They also did better with tolerating side effects. They had demonstrated gratitude towards donors and clinicians, and they were more fearful of consequences. They also had help from others to assist them with medication management. Those who were non-adherent and not taking their medications often had other things going on, life events, they were forgetful, they had side effects that they really did not like, or they couldn't afford or access their medications. And then we have these patients who are somewhere in the middle. They're the patients who often are manipulating their medications, they're a little bit forgetful here and there, or varying their time of their medications. Even though they're taking them, they're just not quite taking them exactly right. Yesterday, Dr. Mannon mentioned the AST drug development conference with patients. At that conference, they also spoke quite a bit about adherence, and patients provided their input on what they felt were challenges to immunosuppression adherence after transplant. And the most common factors that they brought up were listed in this slide. Frequency and the amount of medication, so feeling that they're taking medications too often. We're giving them very specific instructions about how to take their medications with diet and how practical that is. Burden of clinic visits and monitoring. Our patients are often coming back twice a week for the first couple weeks after transplant. The impact of side effects, which we've talked about, as well as the importance of taking these medications promptly. So when we meet with our patients, we're urging them to try to stay within a 15-minute window. I mean, in reality, how practical is that? Ideal, but may not be practical. Patients also provided their input on what they felt adherence interventions should be focused on and how they should be implemented. They spoke to the need to tailor the approach to the individual and not use a one-size-fits-all approach. They offered suggestions on simplifying immunosuppression regimens, and they suggested programs think about patient support groups, using social media and having potential mentors for each other. They also commented on the issue of cost and that having a significant impact, as well as suggesting incorporation of these types of interventions into clinical care. But they acknowledge that there are some significant limitations to implementing these interventions in clinic given the work volume and limited time with providers at times. So now coming back to these 5 dimensions of adherence, what do we think we could do in the pre-transplant setting, and how can we help our patients before transplant to intervene and hopefully prevent issues with non-adherence post-transplant? I've highlighted the areas in blue that might be able to be intervened on pre-transplant. These may include poor aftercare planning, so ensuring that patients actually have an appropriate aftercare plan. Talking about medication education, providing education up front, and ensuring that patients have an appropriate reminder system or organization system for handling their medications. And talking about non-adherence, identifying what the barriers are to adherence pre-transplant and trying to implement a system to getting the patient back on track or using a tool that will help them remember to take their medications. And what about post-transplant? Medication costs we'll talk about in the next couple of slides. Development of comorbidities. This is an area where we can intervene to change immunosuppressive regimens to medications that are a little bit less toxic, removing steroids or converting off of calcineurin inhibitors to less neurotoxic medications. And complexity of medication regimens, trying to simplify routines or regimens, and then side effects, as I mentioned as well. So when we're talking about cost as a barrier, there are a number of tools available to our patients to assist with medication cost. There are 30-day free drug vouchers for a number of products that I've listed here. These are usually just one-time, once-in-a-lifetime vouchers that can be used in anybody, even if they have Medicare. This is what we often end up using if patients run out of their medications and we need to get them something right now. They can usually email this link to the patient and they can print it out and bring it to their pharmacy or just bring it to the pharmacy on their phone. There's also patient assistance programs for a number of medications, Valcyte, Cellcept through Genentech, and then through Veloxis, and Varsis has a really nice program as well. Both of these up on the top are a little bit more liberal with their financial requirements. I've felt like we've been able to get the vast majority of patients into these programs if they're not able to afford their medications. Novartis and Pfizer also have programs as well. And then there's copay assistance programs, but these here are all really limited to the patients who have commercial insurance. But again, something really important to keep in mind. I mean, even just within the past few weeks, I saw a patient who was switched off of Zortress to sirolimus because his copays were too high, but he actually had commercial insurance. So he was somebody who would have benefited from just staying on everolimus and using a copay card, and he wouldn't have to pay anything for his medications. But if you're not aware of these kind of products, it makes it a little bit more challenging. But again, these are just dedicated to patients who have commercial insurance. What about modifying regimen complexity? Thinking about our patients, we often send them home with multiple different medications. They leave the hospital sometimes with a bag full of 8 to 10 medications. If we think about just simplifying at least one, or tacrolimus, if we send them just with ones rather than ones, 0.5s, and 5s, might make life a little bit easier for them, at least in the first couple weeks after transplant. Ensuring that we try to use or minimize use of medications that are all for the same indication, so trying to maximize our blood pressure medications before adding on multiple other medications, and also ensuring that our patients understand why they're on multiple medications for their transplant, so they don't feel that they can just stop one because they have other ones as a backup. And then frequent dose changes, we want to keep in mind with medications that have long half-lives, that if we change a dose on Monday for, say, our Zortress, we don't necessarily want to make the patient go back and get another level on Thursday, because it's probably not going to be something we would act on anyway. What about adherence interventions? There's a number of different intervention types and categorized in various ways. So educational interventions, these are the things that all of us as pharmacists are doing usually at the time of discharge. We're providing didactic, verbal education to our patients about all of their medications. Behavioral interventions, these are usually things like reminder systems, pill boxes, alarms, contracts in some places, and some other places have used peer mentor support groups to assist with this. Organizational, we just talked about the regimen complexity, and then cognitive behavioral or motivational interviewing. This is something that I often like to do with patients, to talk with them about their routines and get them to really explain how they manage their medications rather than asking specifically, do you take your medications, yes or no? If I can get them to tell me a story, I can better understand their rationale for why they're doing what they're doing. And then technology-based interventions, including wireless pill bottles and apps, which we'll talk about. So depending on your patient's level of tech familiarity, you might be able to use apps, or you might have somebody that's just comfortable with a grid on a piece of paper. So the first image there is just your typical chart where a patient might want to write down the medication name and just check the box if they took it. That's for somebody who's very basic. Next, you have your wristwatch. This is your easy Amazon $35 Timex that will vibrate if your patient has a simple watch, or you can go as fancy as your $500 iWatch. Most everybody has an iPhone, and you can easily set alarms on your iPhone to have your patient remember to take their medications if they're not up for a fancy app. There are automated alarmed pill boxes as well. And then there are the glow caps that beep and alert and remind patients to take their medications. And then the information from the cap is then transmitted back to the providers. And then finally, the last one I wanted to mention is the MediSafe app. This is personally my favorite because the MediSafe app has this cool technology where you can add a MediFriend, and the patient can have their support know when they took their medications or not. So if the patient did not click that they took their medications, after a certain amount of time, their MediFriend will actually get an alert and know that they didn't take their medications, and they can then text the patient back or call the patient and say, hey, why are you not taking your medications? This app also has a nice feature in which it allows you as a provider to invite the patient to your portal. So you can see as the provider when the patient has clicked the box to say they've taken their medication. So if you see a trend of a lot of the reds where patients have missed their medications, this can be an alert to you to potentially reach out to the patient about their medication management practices. This app also has the ability to integrate into the Apple Health Kit, so it can pull in vital signs, blood pressures, etc. And it also can link within your Epic healthcare system and pull medications directly into the app as well. So let's come back to our cases and talk about each of them and some potential interventions that might be appropriate for each patient. So case one, this is our working professional having difficulty remembering to take their medications and get to the lab. This person's tech savvy. So what can we do for this person here? So one thing I was thinking we might be able to do would be considering switch to Envarsus, because they're having some difficulty getting to the lab on time. And Envarsus has this 3-hour window on each side, so you have a little bit more flexibility in the timing of your lab. So if they're going to work or they need a little bit more flexibility, that might help. The other thing this person could do is consider adding the MediSafe app to their phone, and that could serve as a reminder. And if they're working and they're at a desk all day, they can have Outlook alerts. I'm not sure if somebody would feel comfortable saying, put your meds in take your medications as an alert, but you can put something like a meeting with the donor's name or something of the sort to remind you that this is an important aspect of your care for your transplant each day. On to case number 2. This is our non-English speaking patient with low health literacy and minimal support. For this patient, we want to make sure we simplify the regimen. First off, we want to make sure we remove supplements that aren't necessary, potentially acid-suppressing medications, PRNs. Try to simplify that med list as much as we possibly can. And then consider use of coded bottles or a coded medication card. There was actually a poster at ATC this year where they used pictures of animals on the med card and on the bottles. This is more geared towards pediatrics, but it's something that everybody can recognize what these animals are. Some people can't see colors. Those colors could work or could not work depending on your visual issues. But colors using the little dots, we've used those by putting them on the med card, and you could use a marker and color the top of the bottle as well. I had another patient who was similar to this patient, non-English speaking. They used a number system where they numbered the medications on the med card, numbered the bottles, and also used the little signature flags that you put on a paper to tell somebody to sign something, and they put those signature flags on the bottles as well, and those colors of the signature flags corresponded to the times of the day that they had to take them. So they had blue, yellow, and pink signature flags. With on the signature flags, they had the number of pills they had to take each time. They were pretty good with keeping that up to date, but that also kind of helped them better understand what they were doing. And then case number 3, we have our patient with significant medication-related side effects, headaches, tremors, nausea, on-tac, immediate release, and mycophenolate. And this is a patient, given their neurotoxic side effects, we may consider switching over to Envarsus or belatacept to alleviate these side effects. Also, you can potentially consider switching the Cellcept over to mycophenolate sodium or everolimus to assist with GI toxicity. And then finally, case 4. This is our patient who can't afford their medications. They lost their insurance. They now have a rejection, and they had a history of being CMV positive to negative, requiring use of thymoglobulin, so likely having to recycle their Valcyte as well. So things to consider with this patient would include enrollment in patient assistance programs, so consider conversion to Envarsus and Myfortic, just given the support that we know the Veloxis program has. They also have that 30-day voucher that you could provide the patient to bridge them until their medication is approved by their insurance. Would enroll the patient in the Valcyte Patient Assistance Program. That program usually will supply a 3-month supply of Valcyte. And if your hospital has a hospital-based patient assistance program, I would also consider using this. A number of hospitals that have 340B pricing have their own internal patient assistance programs where they offer discounted medications to patients who do not or cannot afford their medications. And then finally, I wanted to mention the GoodRx coupons. I don't know if any of you have ever seen that website where you go to the website, plug in the drug name, and it'll give you a cost of the medication at all the various pharmacies, often much lower than what they would typically pay. They just have to print out the coupon, bring it to the pharmacy, and they usually can get a discount for their medications. So in conclusion, I'd like to leave you with a couple practice tips and encourage you going forward to think about adherence as potentially your 5th vital sign. How would you go about doing this? Truly asking questions, asking open-ended questions, and having the patient tell you a story. Other things you can think about is use of technology. I know a number of hospitals are now using iPads in their waiting room, so something we've been considering is potentially adding the BASIS questionnaire onto the iPad for the patient to fill out when they're in the waiting room, and then having that BASIS questionnaire translate into their medical record for other providers to be able to see how the patient is recording how they're taking their medications. I'm not sure if patients would feel a little bit more comfortable doing that electronically rather than having to admit to a physician or a provider in person, but something to consider. You also want to take the time to understand the patient's specific reasons for medication non-adherence. We talked about multiple different risk factors for non-adherence, multiple different interventions. So you really need to understand what it is that's driving the patient's non-adherence. Is it cost? Is it side effects? Is it that they don't understand? Because your approach to each patient is going to be different based on what their issue is. And then finally, I would just want to encourage everybody to use the various members of your multidisciplinary team. Your pharmacists, your social workers, your financial coordinators. We're often very involved up front for the first couple of weeks to couple months, but when patients get much further out after transplant, we're not usually seeing them as frequently. So I just want to encourage you to make sure that you're thinking of everybody that's part of the team that can help you with various needs of your patient's adherence going forward. Thank you for your attention. And we'd be happy to answer any questions.
I think there are just a couple questions before we go to break, and I think that this is actually one of those incredibly important topics that we spend a lot of time talking about, but really frustrated in action. Think about all the drug development that we have to create the next BID drug and all the money and resources that go into that, and then have a patient population there, 35% are not adherent. So if we really want to look at outcomes and look at long-term function, this is really an important topic. So one of the questions is, does a single payer system, are there data looking at single-payer systems versus other systems in terms of adherence?
I honestly don't know the answer to that question off the
I'm
top of
not
my
sure
head. I'm
either.
not sure. Do you know? I'm sure there is, but I don't know.
Yeah.
the answer.
So there are a lot of questions about the MediSafe app. So is it
Yes.
free and
It is free, yes.
And it is something that is able to be downloaded or information downloaded into Epic or some other
Yes,
health
so it will
systems?
link through the Apple Health Kit. So you have to agree to let the Apple Health Kit communicate with your gateway, at least for partners, for us, it's the gateway. And then from there, it will import medications. One of the limitations in that is that it imports all of the medications. So it doesn't necessarily pull in everything that is active right now. So the patient then has to go sort through that and determine which medications that they want to keep as active. They do tell me that it does push medications as well. So when you add new medications, it will push new medications. And they are willing to work with transplant centers too to develop transplant center specific needs. So of course that costs money,
Right.
but they are a great company to work with and are very invested in patient adherence.
Outstanding. Okay, so we're going to take a 15-minute break, and then we're going to be back for Wheel of Knowledge. Okay.
Hi guys. Oops. Hello, hello, hello. It's good. All right. Hi everyone. We're gonna have to move you guys to the front to make the teams. So we're gonna do teams per row. So we have to fill up every row. So this is the wheel of knowledge. So we have a bunch of questions covering topics from immunology 101 to history of transplant to miscellaneous and so forth. We try to mix it up, things that you learn or hopefully learned in the past 3 days. And if you get points if you get an answer right, but you also get points if you get your answer wrong. If you don't answer, you don't get any points. For sure, you get more if you answer right, so it's about 100 points. And if you answer wrong, you get 5 points. But you have to answer so that we can capture that. So let's try to move it. You guys move to the front so that we can make the rows with everyone. So otherwise, we get some teams that are going to be not as favorable in terms of points. And we'll see how many rows or teams we will have total. So do you have to kick us out of these rows? No, let's see, because this row is a little bit empty. Maybe I'll get you guys to sit on, or let's see if they come in and then we'll fill out some of the fronts. So we have split faculty on it. Do you want to count on every line just so that we have it, or about 2, 4, 5? Once you get your seats, just log into the iPad again, and we're going to just do a head count to make sure we have teams that are equal. But I need everyone to come to the front and get your seats so we. But they can vote as well. Yeah, or they should, or yeah.
It's up to you.
Okay.
Does that work? Do these two
Yeah.
as one.
Okay.
2, 3.
Okay.
Does that work?
Yeah. All right. So 1, 2,
This will be team 1.
3, 4, 5, 4, 5, 5.
This is team 3 down this row. Team 3.
Yeah, exits, seat belts. Guys, I think I'm going to need you guys to move one because you got 4, 5, 6, 7, 8, 9, 10, 11, 12.
Yeah, team 4. That's okay.
Yeah?
Yeah.
Okay. That's fine. Yeah. Maybe if you get some. Forgot. Grab him. Forgot his name. Let's see. 1, 2, 3, 4. So how many we got?
Yeah. So we have 5 teams.
5. Okay.
Teams total.
5 teams, guys. Maybe anybody comes in, they can sit on that. Yeah. Because otherwise they will be negative. All right.
Okay.
Are you guys ready? Are you guys ready? Yeah. Come on. So we have the team. So you can raise your arm, your hand. So team 1. Come on, guys.
Hold on.
Team 1. All right. Team 2. All right.
Hold on. How do you know who? I thought it was row by row.
It's row.
No?
Just this one is short, so that counts in 1.
So
Yeah.
this is a group? Okay.
Yes.
All right.
Okay. Team 2. Sorry. You want to be like, you're 1. Yeah. Team 3. All right. Team 4. All right. And team 5. Okay. If you could join team 5, that would be great. You guys as well. Team 5, please. All right. Do we have music or what's going on? Or DJ?
Have you guys found who the ringers
Wheel.
are yet?
All right.
Yeah?
Okay. Hard question. Hard. Mary Jane? So coronary allograft vasculopathy. In the 2010 ISHLT consensus definition of coronary allograft vasculopathy include all the following features except intravascular ultrasound, restrictive physiology, angiographic lesions graded in severity using the CAV 0 to 3 grading, or ejection fraction.
So is there whispering that's going on in between the teams? Can you vote along with your teammates, or should you be separate and distinct?
Usually, you know, vote on your own, but for your team.
But this is kind of like soccer. Everyone gets a prize for voting. There is a 10-point prize for even voting, so don't not vote. You have to vote, and you get a little credit, okay?
All right. You guys can, yeah, of course. All right.
The ringers.
So, all right, Mary Jane, do you want to comment on? You have a microphone there.
Hi. So the answer is intravascular ultrasound is not part of the definition. The rest are.
All right.
Even though that's
So 1/3. We'll see the score in a little bit. Let's go to one more. All right. Histocompatibility. All right. That should be fresh. Okay, so HLA antibody characteristics associated with resistance to desensitization and increased antibody-mediated rejection injury include high titer, cross-match positive DSA, HLA class 2 specificity, IgG3 subclass DSA, complement activating DSA, or all of the above. All right. We should get 100% on that. Otherwise, we'll have to fire Annette. All right. 88. Okay. That was good. That was good. If you hit like 85 and above, that's wonderful. Okay. Next. Immune suppression.
Okay.
Cool.
Oh, yes.
Oh, you want to read it?
Sure. There's a little history. Who is the transplant pioneer who made this following comment? The surgical side of transplantation of organs is now completed. We're now able to perform transplant of organs with perfect ease and excellent results from an anatomical standpoint. All our efforts now must be directed to the biological methods which will prevent the reaction of body against foreign tissue, allowing the adapting of homoplastic organs to their host. Who said that following statement? Alexis Carroll, Joseph Murray, Sir Roy Kahn, Tom Starzl.
Tom Starzl. All right. Oh.
All right. Hold on. You should go back and see this. So do you know who he was? He actually was the first person who he invented essentially the first vascular suturing technique, and he also invented the first storage of organs. So the others were incredibly important as well, obviously. Joseph Murray did the first transplant, so you think surgical side must be perfected. Tom Starzl, of course, did the first liver, and you would have imagined that he would have said such a statement to say, we are now done with the surgical side. And then Sir Roy Kahn actually was the first, he was also a surgeon at Cambridge. Campath came from Cambridge under his watchful investigation, but he also introduced azathioprine as the first immunosuppression agent other than corticosteroids. So,
all right.
Yeah.
Can we look at the score? Just checking. All right. Team 3 is winning.
Team 3.
124. All right. Wheel of Fortune again. Let's do it. All right, infectious disease. Where's our ID people here? So for the organ transplant recipient, the risk of infection is related to the quality of the organ transplanted, past or present infections in the donor, past infections in the recipient, intensity of immune suppression, all of the above. Yeah. Yeah, all of the above. Oh, sorry. Yeah. All right.
That didn't separate money from Team 3 is still in the lead, I think.
All right. Okay, next one. Let's try to get a harder one now. Ooh, lung.
Jeff out on that.
So name a systemic disease which often recurs in the lungs after pulmonary transplant. Idiopathic pulmonary fibrosis, sarcoidosis, cystic fibrosis, primary pulmonary hypertension, or Goodpasture syndrome. Yeah. All right.
That's a good question.
Do we have any lung faculty here?
We do.
Where's Jeff?
He's right over here.
Nice. Jeff, do you want to comment on?
There are actually two diseases that commonly recur, that frequently recur. One is sarcoid, and the other is lamb aphasia, leiomyomatosis. There are a couple case reports about rare disease, but those are the two diseases.
Sweet.
All right.
Learned something new.
Next. All right. History of transplant.
History.
How long did the first heart transplant performed in South Africa last? 7 days, 14 days, 18 days, or 28 days?
Depends upon savvy test takers.
All right. 18 days. Yeah. They're good. So yeah, so it was extremely short, the first heart transplant.
They learned how to
Jane
take tests.
can comment on it, but I thought it was amazing. Number one, that was done in South Africa, but he learned all the technique in the States, and when he went back, and I think this exactly the DCD kidney.
Only in South Africa was there a recognition of brain death. I thought was this story. Everything else was DCD before, but they actually said in South Africa allowed brain death to be a definition of death, and that's how they.
And there was a lot of failures, those initial heart transplants. Even though he gets all the credit about doing the first one, there's so many patients that only survive like a week or two. So
the United States actually, we changed our definition of death right after this because we were beaten to the punch.
Mm-hmm.
Those were all, again, that was, there had been a track record of failed liver transplants up until then. Yeah.
Mm-hmm. All right. Next, let's throw the wheel. Okay, liver. So a 47-year-old male underwent a deceased liver transplant 5 days ago and is not waking up. He's on tacrolimus and his hemoglobin and platelets are dropping without explanation. What is your next diagnostic test? EEG, CT of the brain, a peripheral blood smear, lumbar puncture, ammonia level, I don't know. Consult neurology. Perhaps would be good, yeah. All right, and the answer is peripheral blood smear. All right. So you were worried about thrombotic microangiopathy affecting, especially with the drop in the hemoglobin and platelets. Any other comments, Jamie, from your side?
No, I mean, it's interesting.
Do you want to?
Yes, was not me.
I assume that he was looking for cystitis.
Correct. Yes. And I asked him why it couldn't be ammonia, but in theory his kidneys were coming up. I think a very interesting point that I think as a transplant physician, you should always know there's this hyperammonemia that can happen after, due to infection. So if somebody's colonized by some of these urea kind of breaking bacteria, and it can happen very early after transplant, and the patient becomes like unconscious or in coma like very quickly. But if you catch it early by measuring the ammonia level, you're actually able to start like dialysis and treat the infection and get that resolved. It was mostly in lung that they reported. We had just a case in kidney as well that the ammonia level was like 500. But we diagnosed really quick, so there was no cerebral edema, because that's the major danger. So if you wait for a little bit to diagnose, then the cerebral edema happens, and then it becomes irreversible. But that's an interesting point as well. Okay.
Yeah, I was just going to say, we have seen some
In kids.
Kidney.
refractory ascites, and they'll have really enlarged spleens and other things. So sometimes, and I wonder if that's what he was getting at.
Got it.
Right, and that hence the not waking up.
Mm-hmm.
Transport organ.
Got it. All right. Alex?
Kidney. In a paired exchange program, which pairs have the best match rates? Pairs with blood group O donors, pairs with blood group O recipients, pairs with blood group O AB donors, pairs with blood group A donors.
In a pair exchange program. Okay. All right.
Not bad.
Very good.
I know that, I mean, essentially you'd think, oh, well, AB donors, or I mean, AB recipients, no, never mind. I'm not going to even go down there. I was looking over at our resident desensitization paired exchange person for guidance, but I'm going to leave it alone. Yeah. This is directly out of your slide, I think, from your, yeah.
So blood group O donors, universal donors. So if you bring a universal donor to the table, you're more likely to find a match. If you bring an AB donor, that is definitely not a universal donor, and in fact will only match another AB. So those donors you typically never find match rates.
And typically, if you have an AB, where I was going was AB recipient. If you have an AB recipient in paired exchange, there's a problem, right?
An AB recipient with an O donor is lovely.
Yeah.
All right. Next. Pathology. Not that important. All right. So what's your pathology diagnosis? Recurrent disease, transplant glomerulitis, transplant glomerulopathy, antibody-mediated rejection, and T-cell mediated rejection. It's a lung. No, I'm kidding. And the answer is antibody-mediated rejection.
Should we
You
go
wanna
to the
comment on?
scoreboard?
Huh?
Okay, so you want to comment, or do you want to go?
Yeah, C for D is positive and you had microcirculation inflammation. That's why you could render a diagnosis. And it's not just transplant glomerulitis or glomerulopathy. And not recurrent disease. You should not have C for D as recurrent disease.
I think we should go to the scoreboard.
All right, let's look at the
Who's
score.
winning? Where do we stand on the scoring?
Team 1. But it's actually, it's tight. It's tight. 468, 452, 444.
That is
Yeah,
a
yeah.
tight race. Okay.
The winners will get a weekend in Hawaii? No?
Yeah. I think
Exactly. Sponsored by the AST. All right, let's go again to our wheel of knowledge. Infectious diseases.
cytomegalovirus infection is important in transplant because it's rare, contributes to the risk of opportunistic infection and rejection, cannot be easily treated, people with CMV cannot receive organ transplants, or CMV causes cancer.
All right. You guys are sharp. Exactly. Next one. Oh, Immunology 101. What is the main mechanism of action of belatacept? It inhibits mTOR, it suppresses purine metabolism and DNA synthesis, it blocks CD25 receptor, it inhibits B7 CD28 signal, it blocks the CD52 receptor. All right, 90. Okay, should have been 100. Come on, guys. So yeah, I mean, mTOR inhibitor, the first one. The second is all the anti-proliferative agents, azathioprine, mycophenolate. It blocks CD25 receptor, that's Similac. And the block CD52 is alemtuzumab. Actually, it's a depleting antibody, binds and then depletes the cells. Cool. All right. Next one. Miscellaneous. All right.
Tricky.
Frank Oz, who voiced Miss Piggy, also voiced which famous movie character? That's like Tweety Bird, Yoda, Popeye, or Cinderella.
This is what's going to separate the winners from the losers.
That's Carly. All right. Let's see. Yoda. 52. Okay. Can we look at the scores again?
Team 4.
Team 4. we got some rotation there. I like it. Team 1 dropped, guys. Come on. All right. Yeah, I don't know. It's secret, yeah. Lung.
Risk factors for primary graft dysfunction include all of the following except donor cigarette smoking, positive sputum gram stain, undersized donor relative recipient, donor heavy alcohol use, aspiration.
I assume this applies to lung.
Risk, yeah, it's a
Lung
lung
transplantation.
question.
Yeah.
Lung category.
All right. We'll look at the answer. Wow.
Woo-hoo.
Jeff, we need explanation
That
there.
was a good question. Here.
So this comes from sort of collected data, but it's from the ISHLT consensus conference, and basically the positive sputum gram stain really doesn't seem to predict primary graft dysfunction. The biggest one, the one that most commonly seems to come out, is donor cigarette smoking.
Okay, nice.
Donor cigarette smoking.
Cool. Have it.
All right. Next one.
Da-da-da-da.
All right. Let's turn the wheel. Liver. Okay. A 74-year-old female underwent a liver transplant 1 month prior. Develops a new skin rash, cytopenia, and diarrhea. Liver tests are normal. What is the diagnosis? CMV infection, drug toxicity, graft versus host disease, PTLD, or S. myasthenia? We don't know. All right. Graft versus host disease. Jamie, do you want to comment on graft versus host? Something we don't see in
Liver
kidney,
graft versus host.
but
How often does it happen?
So in my whole career, I've had 3. One was this year. I don't know, that's just an anecdote. I don't know off the top of my head. Annette, do you know how often It is much more often in multivisceral. So if you have intestinal, liver, in fact, Cleveland Clinic. Anyone here from Cleveland? You.
Yeah.
Really?
So Cleveland started a protocolized testing for donor-derived cells in the patient's blood, so chimerism, following multivisceral transplants, because you actually pick up chimerism quite often. That is a very frequent in a multivisceral transplant. However, actually developing graft versus host is rare. But as Jamie said, sometimes by the time it's diagnosed, it can lead to high mortality. And the other thing is your chimerism labs, they don't understand stat testing. So we've had to step in as the HLA lab and do some quick HLA typing of blood to try to pick up, and actually we even HLA type the skin to give it a more specific confirmation that it is in fact donor lymphocytes in the skin lesion.
We currently have a patient who's doing very good multi-organ, multi-visceral transplant, and his current chimerism went from 10 to 20 to 60%. And everybody's getting alarmed and is going to be listed.
Yeah, so we have a rising chimerism in a.
What's the chance of really develop full-blown graft versus?
You know, graft versus host disease, it's a fine balance between how over immunosuppressed the patient is and how rigorous those infiltrating or the incoming lymphocytes are.
But it doesn't just mean that the organ lymphocyte overwhelming the system
Yes.
more than the patient own lymphocyte.
Yes.
All right.
Unlike bone marrow, you have to lower immunosuppression of that one.
Actually, I think we've they might treat like with Campath and just kind of do a lymphocyte depletion. At least they were treating with thymo, and I know other centers have, because it's a T-cell mediated disease, so they treat with thymo or Campath and kind of just get rid of the
donor lymphocytes.
the donor. Yeah, they take
Yeah.
out the recipient, but they take out.
Yeah, like this.
I do believe photopheresis has been used as well.
So it was the rash the distinguishing factor in this question?
Yes.
I mean, yeah.
So
When you find
CMV
donor-
tends not to cause any rash, so I think that CMV kind of drops the diarrhea for sure.
Okay.
And yeah, CMV can have all these other obscures, but at least skin is not made one. Should we spin the wheel?
Let's
Yeah.
spin one more, and then we'll
look
go
at scores.
more.
Okay, heart.
Heart. After, okay. All of the following events directly and negatively impact short and long-term survival after heart transplant, except development of de novo DSA, development of chronic allograft vasculopathy, CMV infection after transplant, pre-transplant ECMO support. Directly and negatively impact short or long-term survival.
It's a heart one. All right. Let's see. CMV infection. It does not affect.
I mean, in large registries, it's viral infection, but non-CMV. You can't actually make that association in large registries, even though there's tons. That's what we truly believe, and probably CMV, vasculopathy, and death. But
Interesting. I think for kidney, it's definitely.
It is it's associated in kidney. It's
Yeah.
associated with
Yeah.
graft loss and mortality,
Yeah.
of course. But again, it's one of those associations, but the direct effect. Have you, Michael Mengel, have you ever seen CMV nephropathy? No. I haven't either, and I think that this is something that is, something that's CMV in the kidney. We're always staining for it, and I've never seen it in 20 some odd years. You haven't seen it in 20 some odd years. Yeah.
I've seen one.
You have seen
Yeah.
CMV staining in the kidney.
No, but it was. Yeah. Well, it was look at like a microscopy and then confirm on the endothelium. Yeah. But I think one thing about the CMV, which I think is extremely interesting, among all the infections you can have after transplant, CMV is the one that kind of changes your immune system the most. And if we were talking about like the fingerprint of the T cells, the TCR clones, that we have all these, multiple millions of clones in our circulation. If you had a CMV infection, about 30% of our clones actually recognize CMV after you had an infection. So it kind of switches things, in a tremendous way, and it's very unclear why a single virus had such a powerful effect. But we do know that they impact tremendously, I think, the immune system in the long run.
Okay.
All right.
Let's take a look at the score, shall
All
we?
right, let's do it.
Okay.
Oh, team 1, 800. It's tight.
Look at that.
It's tight. It's tight. All right.
Team 5, man.
Team 5, we need you guys back. Yeah.
You gotta bring it
What's
up.
going on? John, please join Team 5 there. Immune suppression.
Immunosuppression. Which immunosuppressive agent received FDA approval in the absence of a phase 3 randomized controlled trial? Mycophenolate mofetil, thymoglobulin, sirolimus, belatacept. All right, good. So again, we talked a little bit about that in the group session yesterday, but basically there were two very disparate studies that were then combined and re-reviewed and reanalyzed that ultimately the FDA accepted after a lot of, many years, following the study. Actually, about 10 to 12 years after the studies were done. So.
All right. Let's spin the wheel again. Oh, history of transplant. Cyclosporin was extracted from which organism? A virus, a bacteria, a plant, a fungus, or an alga? Oh. That's a good one. All right. 90%.
Nice.
Fungus.
In Norway, it was piping for fever diabetes.
Oh, really?
Huh. Interesting. All right. Let's spin it again. Immunology 101.
Okay.
Okay. The basic immunoglobulin structure is composed of 2 identical heavy and 2 identical light chains, 2 identical heavy and 2 different light chains, 2 different heavy chains and 2 identical light chains, and 2 different heavy and 2 different light chains.
Can you read that again?
All right. So it's 1. So actually, when you produce your antibody, it's going to have equal, the same kind of light chain. So it's either lambda or kappa. For sure, we have cases where you can produce those light chains and without the heavy chain, and you can even have some of these that can be toxic. But in general, it's an identical. And this is the way also that, it kind of helps you identify like a proliferative disease, because it's usually a single clone that's responsible. All right. Let's do it again. Histocompatibility. All right.
Do I have to read that?
Yeah.
Typing of which donor HLA loci below is necessary to determine the presence absence of donor-specific HLA antibody, DSA. Typing of which donor HLA loci? HLA A and B, HLA A, B, and DR, HLA DQ A1 and DQ B1, or all HLA class 1 and class 2 loci?
All right. Annette, jump in.
Sneaky.
If you want to know if there's HLA-specific antibody to your donor, you must know the typing of every single loci. Both the alpha and the beta chain. You can have antibodies to just the alpha chain, just the beta chain. And there was a question in the online about HLA-C. HLA-C antibody, HLA-C and HLA-DP tend to be less immunogenic. We get fewer patients with high titer antibodies to those. However, I have seen at Hopkins an HLA-C DSA that was strong enough to yield a positive CDC crossmatch, and we certainly avoided it. So
All
And I
can
think,
be.
yeah, and historically, why AB and DR is what most centers kind of use, because that were the most polymorphic and, where they were doing initial testing. But now, start to expand even further with the DP and C and so forth. All right, should we look at the score? That was, yeah.
One more. Let's do one more
Probably going to
question.
split the teams there.
Let's do more questions.
Huh?
One more question. Let's see if.
Okay,
someone
one more.
can count.
All right, one more. Miscellaneous. Okay. When did the American Society of Transplant Physicians change their name to the American Society of Transplantation? 97, 98, 99, or 2000? You cannot Google, okay? You're good? All right. Awesome. Then we'll, okay. Calm down.
98. Oh my
All
God.
right.
Wow. I would have missed that one.
Yeah. Okay, should we look at the score?
Yeah, let's go to the score.
20%. Team one. All right. Okay, we are in our last stretch, so let's spin the wheel again. Lung.
Oh gosh.
Okay. Which gram-negative bacteria species is associated with poor outcomes after lung transplantation for cystic fibrosis? Burkholderia pseudomallei, Mycobacterium abscessus, Pseudomonas aeruginosa, Burkholderia synopsistia, Burkholderia something. Burkholderia multivorans, yeah.
There's got to be some infectious disease docs here too, right?
Ah!
Wow. 50% got that right.
I was chosen Pseudomonas. Oh, Jeff.
So CF patients are colonized with a lot of bacteria, and historically, before we had sort of better speciation, it was Burkholderia cepacia sort of group, and then when they were subspeciated, Burkholderia cenocepacia turned out to be the one that is really associated with poor outcomes. Burkholderia pseudomallei is not an organism that that is really associated with CF. It causes melioidosis. Mycobacterium abscessus is also associated with poor outcomes, but it's not a gram-negative bacteria. And the other two, aeruginosa and multivorans, are seen in CF, but are not associated with poor outcomes.
All right. I would have guessed Pseudomonas initially. All right. Let's spin the wheel again. Yeah, exactly. Kidney.
What genetic variant is associated with increased risk for kidney disease among individuals of African ancestry? CYP3A, uromodulin, ABCE gene, apolipoprotein L1.
I don't know. Yeah. I hope he does.
It's going to separate out the kidney.
All right. Awesome.
That's about time. Okay.
Yeah. Well, that's the most important gene that was discovered in kidney disease, so I'm glad you guys are, yeah. Oh, and, all right, let's spin it again. Heart. Okay, which donor future reported to be associated with worsened post-heart transplant outcomes? Low ejection fraction, downtime, troponin levels, donor age, or donor recipient predicted heart mass ratio below 30%? Which donor future?
I like it. It's a good question.
You have your Jane. Can you press it? Oh, that was not good.
Sneaky.
It's
Jane?
It's definitely donor age. Can I tell you, that is the most robust predictor and ischemic time.
Sleep.
So low EF doesn't predict, downtime doesn't predict, it's not accurate. Even troponin doesn't predict, and the predicted heart mass ratio less than 30% is what you are going for. So greater than 30% would be a problem.
Okay.
Great question.
So if you ever need a heart, you want to have a young one.
Great, yeah, you don't want too big of a heart.
Yeah. Okay, let's spin the wheel again. Histocompatibility, all right.
Calculated panel reactive antibody, CPRA, provides HLA sensitization data regarding strength of HLA antibodies, likelihood of finding an acceptable donor, number of HLA antibodies per candidate, reflects all HLA antibodies detected greater than 1,000 MFI.
Ooh, there are two there. This is like a quiet one. I think everyone's like, psh. All right. Yes.
Good job.
Yeah.
97%.
All right. Okay. Next. Immune suppression.
Okay. What target of co-stimulation blockade resulted in thrombotic events in clinical trials? CD40, CD40 ligand, CD8086, PD1, CTLA4.
CD 154 is. Yeah. Yeah. All right. Oh. 1/3.
So that was, I mean, there's newer iterations, but this is the trials that Andrew Adams was talking about in non-human primates, as well as in lupus trials and in early utilization of CD40 ligand or CD154 targeting.
Yeah. And it was a very powerful agent, like in mouse models, in animal models in general. So people had a lot of excitement on this, but when the thrombotic events happen, now the reiterations with the CD40, we have to see the data coming out.
Yeah.
Yeah.
Cool.
All right. We're, last few questions, so now you have to be sharp. Do you want to look at the score first?
How many questions do we have left, approximately? Well, how many more do we have? Six more.
All right.
We should look at the score.
Let's look.
Oh!
Whoa! Tie, Team 4 is back. Team 4 is back.
Nice.
Okay.
6 questions to
All
separate.
right. Infectious diseases.
Fever in organ recipients is always a sign of infection, is usually a sign of graft rejection, must always be treated with antimicrobial agents, is often absent during infection in transplant recipients, is a sign of antimicrobial resistance.
All right.
94%. Jay would be proud.
Yeah. No. The through is okay. Next one. 5 questions to go. Lung.
We got, oh good. This will probably separate a few people.
Yes.
Lung questions are tough.
Which clinical practice is best supported by randomized at least one clinical trial in lung transplantation? Fundoplication for GERD to prevent bronchiolitis obliterans syndrome prior to lung transplantation, addition of an mTOR inhibitor to prevent progression of bronchiolitis obliterans syndrome, addition of mTOR inhibiting association with reduced CNI to prevent progression of mild to moderate renal dysfunction, addition of an mTOR inhibitor to prevent no-melanoma skin cancer after lung transplant.
Okay.
So they have this big trial came out called Forever Lung, and they use low dose mTOR with CNI, and they have better kidney function. I was talking. I was like,
That was one of the clinical trials you discussed yesterday. So, how many patients How many patients were in that trial?
I just wanted to say forever long again.
Oh yeah, here you go.
Forever.
But it was 130 patients.
And,
so,
and it was forever lung?
yeah.
I love it. Okay.
All right. History of transplant.
History.
Okay, when were the first successful kidney transplants performed between identical twins and non-identical twins?
Ah, that's a good one. This is good.
No Googling. All right, 72, 1960, yeah.
So we
4
must
days later.
We must be down to 3 questions now.
3 questions
Do we have
left?
3
Is that
left?
it? 4.
We're 4. Okay, we'll do one more, then we'll look at the score one more time.
All right. Liver. Yes. Okay, a 60-year-old male underwent deceased donor liver transplant 7 days ago. He has persistent ascites and bilirubin is 10. INR is 1.7, ALT is 300, all improving slowly. What do you do next? Liver biopsy, a liver ultrasound with Dopplers, sit tight. This is delayed graft function. Post-transplant lymphoproliferative disorder, list for retransplant, quit fellowship. All right. And the answer is? Oh! Yeah! All right. Jamie, do you want to comment on that? Just get the microphone.
Betsy?
So he's not here to defend himself.
Yeah, Josh is not here, so you
Josh
gotta.
is not here. We wrote these questions. I think that's a very bold thing to do. I agree, it probably is delayed graft function, but I would not sit tight. DCD allografts are notorious for causing all sorts of issues. I'd want more information. For sure, we would definitely get a liver ultrasound with Doppler just to make sure there wasn't an early arterial complication. We would be concerned about biliary issues. I mean, we would sort of work this up. We probably wouldn't just sit tight. Maybe for a day.
We should record that and send to Josh then.
But we wouldn't just sit tight.
Yeah.
Because if you're wrong, it's a problem.
But then 1% will quit fellowship.
All right.
Yeah.
Let's see the scores coming into the home stretch.
Strong work, Team 4.
Where's team? Raise the hand, Team 4. Woo! Okay,
all right.
Okay, let's see if you can keep the lead.
Okay. Kidney.
For patients with incompatible living donors, what option is associated with the greatest survival benefit? Remaining on hemodialysis, waiting for a compatible deceased donor kidney transplant, crossing the immune barrier, HLA incompatible live donor transplant.
Let's see.
Okay,
All right.
could have been some separation there. 26%.
74.
Okay.
So that was the New England Journal of Medicine paper that compared dialysis or transplantation with transplantation. Dialysis is so poor, the outcome, so it's any small improvement, it seems to affect the survival. Go ahead. Precast. Okay. Fair.
It is, yeah.
Okay.
If the candidate had a 99.5% PRA.
Yes.
You
That is true. That was based on precast data. But in general, dialysis is bad. So there's going to be some sort of cut point, I think, upon which you have waited too long. And so I think when you look at the patients who are achieving transplant under CAS, who are highly sensitized, they often have many, many, many years of dialysis time. So just because you have that doesn't mean you instantly get a transplant. So I think you really have to, I don't think CAS has completely solved the problem for all of those patients. Just saying.
And, well, you also don't know the degree of the positive cross-match, if it's a wildly positive incompatible donor. Yeah. Anyway, I think those are...
All right.
The answer wasn't correct, I think, but anyway. So we got down to 2, right?
Down to 2 questions.
Okay.
All right. Pathology. All right. Your pathology diagnosis. So is this recurrent disease, transplant glomerulitis, transplant glomerulopathy, antibody-mediated rejection, or T-cell mediated rejection?
Just close your eyes.
All right. T-cell mediated rejection. 68%. Michael.
What?
You want to come? Can we go back to the image?
Was there a glomerulus?
I was going to say, I saw no glomerulus.
Yeah, so Lord, how many people hire you when you diagnose transplant glomerulopathy? Stain on the right. Good question. Stain on the right is the granzyme B stain. Cytotoxic T cells causing tubulitis, the hallmark of T-cell mediated rejection.
All right. And then vascular.
Yeah, vascular, it's a B lesion, which you can see a T-cell mediated rejection.
What's the difference between glomerulitis
I think.
and B and C? What's the difference?
Glomerulitis is when you have inflammatory cells in the capillaries, and glomerulopathy is when you have the double contours of the glomerular basement membrane as the chronic stage of that.
Kind of thicken those wire loops. All right. Down to two questions?
I think we have 1 left.
1 left?
Should we look at... Should we...
Two left. All right. So that's is that a question? Infectious diseases.
Okay. Organs from potential donors considered to meet criteria for PHS increased risk for transmission of infectious disease cannot be used. True? False? This probably is not going to separate the winner from the loser, which is good.
Yeah. All right.
Okay.
Last one? Okay. Let's do it. Oh, immunology 101.
Yes.
CD4 expressed on the surface of T cells is essentially an intracellular glycoprotein, is heterodimeric, binds processed peptide in its outer groove, binds to MHC class II on antigen-presenting cells, is highly polymorphic. All right. And the answer? All right. Very good. So remember, there's the T cell receptor, but there's also CD4 that comes along and kind of stabilizes the interaction. So yeah, 86%. Holy cow. Now, what do we do? We look at the scores.
Who's the winner?
All right.
Okay.
And the winner is Team Okay. We'll have to take a picture of the. Oh, my co-chair here.
Oh, yes.
All right, guys, raise your hand, team 4 there. All right, we'll do a
Awesome.
picture later with the team 4 as well.
Nice. All right. Well,
It was a pleasure having you for the past 3 days. So, as we said in the beginning, I think this is one of our favorite meetings. I had the pleasure to co-chair with Alex for the past few years. So this is my last year as chair. So I'll pass it on to Anita, who is going to take over my spot. But I really hope you guys enjoyed. I hope you continue to get excited about transplant, which is one of the objectives of this. Stay in touch with each other and with us. Get involved with the AST. Because I think it's a tremendous resource that you have, and for your career, for networking, for projects and research, for clinical decisions, and so forth. And the faculty here, thank you so much again for coming, for making this.
and please. Thank you. Thank you. Please fill out the surveys. And honestly, and if there are ways to make comments, we really want to try to make the program as robust, and what you tell us can be incorporated in the future. So we change it every year, and we are always looking to make it better. So please help us, okay? And
team 4, you guys are going to take a picture down here.
Yeah.
And get your check.
And another way to get involved is that every COP within AST has a trainee member on the executive committee. You have to be elected, and so you have to be a member of AST by the end of the year. But I really encourage you to reach out to the COP leadership, and if you're interested in being in a COP, it's not an excessive amount of work, and I think you'll learn a lot about AST and about how you can be involved.
Thank you. Agreed. Okay.
Fantastic.
Well, I think that
right.
we
Safe
hope that you have a good, safe travel home, and we're back and better next year.
So we have.
Oh,
just
yes,
to,
we absolutely. Yeah.
Say thank you to Carly and Joanne. Yeah, without them, we couldn't make this happen.
Especially setting up this last event. I mean, this was an amazing.
And we have
event.
our media guys. Thank you.
Yep.
All right.
Okay.
Team 4. Let's do a picture.
Okay.
So yes, go for it.